began in 1992, and the first course was a genetics course through California State University at Hayward, and this was a 1/4 unit under the topic of genetics. I then took a...
Could we just give us a little description, if you would, of the course and how it might relate to the testimony that I'm going to be eliciting from you?
I'm going to object. I think the question was about undergraduate courses, and I think she's talking about after graduation.
Why don't you rephrase the question?
Sure. Have we moved from undergraduate to graduate courses?
We moved from undergraduate to post-graduation courses.
Okay, so the genetics course that you took is a graduate course,
No, that's a post-graduation course.
a post-graduation course.
Exactly. After I graduated from college, it's a course I took after I had already obtained a degree, a Bachelor of Science.
Okay, could you briefly describe that course?
Yes, it was the study of genetics. Part of the course involved DNA and how DNA functioned in the body, and also molecular biology.
Was there actually any hands-on work in that course?
No.
Okay. What was the next course that you took that contributes to the area of expertise that I'll be asking you to testify about?
The next course I took was a 2-semester course in molecular biology, and this was through the University of California at Berkeley and their extension program. And this began in a winter semester of 1992, and it continued through, I believe it was May of 1993. And it was a 2-semester course and involved molecular biology, going into DNA, RNA, and the function of it in the body.
Okay, was any of that work hands-on work?
No.
What was the next course? Or strike that. You say it was a 2-semester course?
Yes.
How many credits, or how many hours did you actually go to class every week?
I believe it was two nights a week for 3 hours, and it was 4 units per semester.
Okay.
So a total of 8 units.
And what was the next course then?
The next course, I was sent to the FBI Academy in Quantico, Virginia, for a 4-week course in forensic DNA technology, both in classroom training and laboratory training. And this was a 6-unit graduate level course. That's it.
When was that?
That was in the month of June 1993.
And would you describe the kind of work that that course consisted of?
Yes, that focused on DNA analysis as it pertained to forensic analysis.
Could you please expand on that a little bit more?
Sure. We went into both RFLP and PCR techniques, and it was both the practical application and also the theoretical application behind RFLP and PCR. We did in-lab practice testing of samples and also some unknown samples by both the RFLP and the PCR methods.
How many hours a day did that course consist of?
That was in excess of 8 hours per day.
And for over how long a period of time?
4 weeks, but not Saturday and Sunday.
And how many credits was that course good for?
It was 6 units of graduate level through the University of Virginia.
Okay. And what was the next course that you took that contributes to your expertise in forensic DNA typing?
The next course was in the fall of 1993, and this was a course, Statistics for Biologists. And it was a 1-semester course through UC Berkeley Extension.
Can you describe what that course consisted of?
That course was on the basics of statistics, and particularly how it related to scientific research, like developing studies to be used in both a clinical environment and pharmaceutical environments.
And how does that relate to the area of forensic DNA typing?
Well, it helped me understand the papers, the literature on statistics and how statistics are used for DNA analysis.
And then what was the next course that you took, the next additional course that you took?
The next course, I'll need to refer to my
Sure.
CV.
Mr. Glazer, do you have a copy of that?
I do not.
Mr. Harmon, do you have a
Thought it
copy you
was-
can show to Mr.?
Sure.
Do you have any extra copies of that?
Yes.
All right, Mr. Blazer, why don't you approach the witness to get an extra copy from her?
This is actually a copy that was discovered by the defense.
All right, Mr. Harmon?
Okay, have you had a chance to look at it, or do you need it?
Yes, I have. And the next course, it's not on Mr. Blazer's copy. It was a DNA sequencing course through the University of Northern Colorado in Greeley, and that was in the summer of 1994.
What did that consist of?
It was DNA sequencing. It went through both the PCR extraction techniques and also sequencing of DNA.
Okay, in addition to those courses that you've described, is there something called the California Criminalistics Institute?
Yes.
Have you taken courses through them which relate to any areas of expertise in the field of criminalistics?
Yes, I have.
Could you describe what, or tell us what those courses are, just briefly describe them?
Okay, as the courses that pertain to DNA analysis were a one-week course through the California Criminalistics Institute, and it was on PCR analysis. And this course was 1 week, 8 hours a day, and it was taught by some of the more prominent members of the forensic community, such as Dr. Edward Blake, Dr. Becky Reynolds, and Dr. George Sensabaugh. And this course was both a practical, meaning we did laboratory work, and also a theoretical course.
And how many hours did that course consist of?
It was a 40-hour course,
Okay, over how long a period of time?
1 week.
1 week. And what was the next course that you took through the California Criminalistics Institute?
Well, that was the most recent course that I took. Prior to that, I took extensive courses in the area of general criminalistics, and I'll need to refer to a sheet. The list is quite long. And these ranged from techniques for conventional serology, such as basic microscopy, and this was a 1 week course, in the use of the microscope as it pertains to forensic work. And also zone electrophoresis, which is a technique used for genetic markers such as enzymes. I also took a sexual assault evidence course, and this was a course that was devoted to extraction techniques for sexual assault cases, examining evidence that pertains to sexual assault cases, and also the analysis of evidence for sexual assault cases. Would you like me to continue on this list?
Would you please? Sure.
Okay. I took a course on analysis of low explosives, and that was in April of 1992. That was just prior to transferring to the DNA laboratory. And that was a course taught by ATF, Alcohol, Tobacco, and Firearms, and that was through the California Criminalistics Institute also. And a course in the analysis of Clan Lab evidence. And this is analysis of samples for clandestine laboratory, meaning drug laboratories where individuals are making PCP or methamphetamine or some other type of illegal drug. I took a arson accelerant course in 1991, and that was also there were some individuals from ATF, Alcohol, Tobacco, and Firearms at that course. And that was on analyzing samples that had been involved in arsons and detecting for petroleum distillates, gasoline, and other substances. In 1990, I took a course in basic serology, and that was just looking at doing presumptive tests on various fluids, blood, saliva, and determining its origin, and also doing genetic analysis or looking at markers such as enzyme markers. Going back a ways now, in 1989, I took a course in crime scene investigation, and that was a 1 week course taught in Eureka by some of the more well-known individuals in the crime scene investigation and reconstruction field, such as Jerry Chisholm and Joe Reinierson. In 1989, I also took a firearms safety course, and doing general criminalistics, it's important that the criminalist knows the basics of firearms. So if evidence comes into the laboratory, they will be cautious and they won't be hurt with the guns by any way. And that was just a 3-day course that taught the basics of firearm examination. And that looks like the extent of my general criminalistics coursework through the California Criminalistics Institute.
Okay. You mentioned that in 1988 you were hired as a criminalist. Could you describe your assignments during the years that you worked at the Modesto Criminalistics Laboratory starting in 1988?
Yes, I started in 1988, shortly after graduation, and I was at the Modesto Criminalistics Laboratory. This is a laboratory that's located in the Central Valley of Northern California. I worked there for 3 years, and my primary duty was crime scene investigation and reconstruction, blood alcohol analysis, and drug analysis. I also did some basic serology at that time. So through the course of those 3 years, those were my predominant duties.
And when you say basic serology, do you mean conventional serology such as ABO typing, EAP, PGM typing?
Yes, at the Modesto Crime Lab, the tests that were done were primarily presumptive tests and also some ABO testing.
Okay, and then did you move to Stockton at some point in 1991?
Yes, and at the end of the summer in 1991, I transferred to the Stockton Laboratory. And at that time, my primary duty was doing conventional serology. And also, as part of the rotation basis, crime scene investigation had to be done at that laboratory also, and also clandestine laboratory investigation.
And are both the Modesto lab and the Stockton labs, are they part of the Department of Justice, the State Department of Justice?
Yes, they are.
Okay, and then in 1992, you moved over to the DOJ DNA lab?
Yes, at the end of the summer, or I believe it was August of 1992, I transferred to the DNA laboratory in Berkeley.
And starting with your initial assignment there, could you please describe the evolution of your role there from when you first started in 1992 until the present?
Yes, when I first began with the Department of Justice at the Berkeley Laboratory, I was assigned to their conventional serology program and getting the protocols in place, and then also doing analysis of samples that from the convicted offender program. The conventional serology program or protocols were put into place. And just as a side, the laboratory decided not to continue on with conventional serology, but to focus with DNA analysis at that laboratory and to allow the satellite laboratories to do all the conventional serology, but to have the Berkeley DNA laboratory focus only on DNA analysis. So initially I compiled the standard operating procedures for the laboratory along with another individual, Donna Dowden, at the laboratory. And I also did 290 analysis. And 290 analysis are samples from the convicted offender felon program. And these are samples when convicted offenders are released from prison, a blood sample is taken from them. And these samples are analyzed and then put into a database in the laboratory. So I did that for the first approximately year that I was at the DNA laboratory.
Just stop you for a second. When you say you did that, what kinds of tests did you submit those samples to?
That was RFLP analysis and PCR DQ alpha analysis.
Okay, and then after that, what was your next assignment?
After that, 2 individuals, myself and Richard Showalter, were assigned to the development and in-house evaluation of a new marker called D1S80. And that began in June of 1994. Richard began the process in June of 1994.
Okay, why don't we stop that for a second? We'll come back to that in a minute. Have you given presentations at various meetings in the area of forensic DNA typing?
Yes, I have.
Could you describe or name the presentations in the groups and just briefly describe the nature of what your presentation was?
Yes, referring to my CV once again. In February of 1993, I gave a presentation at the Modesto Memorial Hospital, and this was for a trauma symposium. The title of my lecture was DNA, the latest crime-solving tool. And this was it was approximately an hour presentation in which I talked about analysis of samples by RFLP and PCR and how it related to some of the trauma nurses and the trauma doctors, the evidence that they had collected.
Okay, and then what was the next presentation?
The next presentation was in Santa Rosa, and that was the FBI put on a crime scene investigation course. It was in 1993, and I was asked to talk about DNA and biological evidence collection.
And then next after that?
The next several presentations were through the California Association of Criminalistics, and one was a DNA study group in San Diego, California, and that was in October of 1993, and it was on the validation of D1S80. And then in 1994, at the CAC seminar in Northern California, I gave a presentation on PCR typing, casework, and courtroom considerations. And in that talk, I talked about both the validation studies that have been conducted on D1S80 and also some of the recent cases at that time that we had done in which D1S80 used in conjunction with DQ alpha gave valuable information.
Okay, now, have you ever testified as an expert witness in the field of forensic DNA PCR typing?
Yes, I have.
How many times?
2 times.
And how long ago was the earliest one?
The first one was it was the latter end of last year, and the second one was in Marin County approximately a month ago.
Okay, and you ever testified as an expert in the field of forensic serology in your career?
Yes, I have.
How many times?
Approximately 5 times.
And have you ever testified as an expert in the field of blood alcohol and controlled substances?
Yes, I have.
How many times?
The combination of the two, approximately 50 times.
Okay, let's get back to the evolution of the forensic DNA PCR marker D1S80 in the laboratory. I think you mentioned that, or strike that. When did the lab first begin the implementation of that PCR marker D1S80?
We first began using D1S80 for casework in April of 1994. That was after approximately 8 months of method development and evaluating the technology.
Okay, now when you say after 8 months of method development and implementing the technology, is this something that the DOJ lab itself invented or put together?
No, it's not.
Okay, had there already been published articles in the peer-reviewed scientific literature about the PCR marker D1S80?
Yes, there was.
Okay, and just before the DOJ lab began this implementation and evaluation of it, could you kind of turn back the clock and tell us what kinds of scientific literature was out there already in existence at the time that DOJ began its implementation?
Well, originally, it was in 1988, an individual by the name of Dr. Nakamura discovered the location of D1S80, and that was published in a journal article. At that time, the laboratory, I believe it was Salt Lake City, continued examining that particular region. And in 1990, an individual by the name of Dr. Kasai published the sequence for D1S80.
All right, Ms. Montgomery, could you spell that for me, please? Dr.
Kasai, Kasai.
Right. Thank you.
Okay, and that's initially. What other sorts of literature was out there in existence already at the time that the DOJ began its implementation of that marker?
Well, the Europeans were using D1S80 long before the United States began using it for casework. An individual in Holland by the name of Sanjantila, I believe that's how you pronounce the name, Sajantila, published some articles on the technique of visualizing D1S80 and also on the PCR technique. And he also published the first paper on casework analysis of D1S80's use for casework analysis. Dr. Badoli of the FBI had published some articles also on D1S80 use, and then there are various other articles that were published prior to DOJ using the technique.
So is there a distinction between a laboratory implementing a marker that's already been validated in the published peer review literature? You see understand the question?
Could you restate the question?
When you implement something, do you implement something that has never been invented before?
No, the D1S80 was examined by a lot of other laboratories, and there was published information out there. And the way the Department of Justice, we looked at the literature, compiled some of the literature, and then did our own evaluation of the technique. And also at the time that we were doing our evaluation of D1S80, we were looking at the Roche. It's a company that a commercial company that manufactures the D1S80 kit. They also had a written protocol for the use of D1S80. So what we did is we looked at all this information, evaluated various techniques, and then came up with a method using the same PCR technique, and so we could use D1S80 and examine it using various conditions.
Now, was the laboratory already using the PCR marker DQ alpha at the time D1S80 was introduced?
Yes, our laboratory was.
Okay. And you know Dr. Ed Blake? You mentioned him.
Yes, I do.
To your knowledge, is his laboratory doing D1S80 work now?
Yes, they are.
Sustained. Curious to disregard that. Proceed.
Are you familiar with an article, peer review article in the journal Forensic Science, generally known as the casework article, where Dr. Blake is one of the authors?
Yes, I am.
Is the PCR marker D1S80 discussed in that article?
I would need to review that article.
Okay, we'll do that at the break. How many cases have you actually worked on where you've used the marker D1S80?
I personally have worked on over 20 cases in which I used D1S80.
Okay. You mentioned that Roche, the company Roche, is this a commercial product that's available through Roche, D1S80 marker?
Yes, the primers are available through Roche.
Okay, and what else has Roche put together to assist people that want to use this marker?
Well, Roche has the full kit. Roche is the company that also puts out the DQ alpha kits. And with the D1S80, they have a similar kit in which they have all the reagents that are necessary for the amplification process available in this kit that can be purchased from them.
And this is the same company that puts out the DQ alpha kit?
Yes.
The same company that puts out the polymarker kit?
Yes.
And are they also, scientists from Roche, are they also some of the authors of articles that are out there about the D1S80 marker?
Yes.
Have you been subjected to proficiency testing in your career at the DOJ lab in the area of forensic DNA typing?
Yes, I have.
Could you please describe the nature, types, and results of the proficiency tests which you've taken?
Yes, our laboratory has a quality assurance manual that states the frequency that proficiency tests must be given to analysts. And for a casework analyst, you have to do 2 proficiencies per year. And to date, I have done 5 proficiency tests, which dealt with over 20, I believe it's 20 samples.
Can you describe some of the nature of some of the samples that have been in those proficiency tests?
Yes, there are both blood stains and sexual assault samples, meaning there's such as a vaginal swab that has both sperm and male contribution sperm and also the female contribution epithelial cells. So I've done both the blood stain analysis comparison and also the sexual assault comparisons.
Have you made any mistakes in any of those proficiency tests?
No, I have not.
Does that mean it's not possible for you to make a mistake?
No, it doesn't.
I believe you mentioned standing operating procedures. Does your laboratory, the DOJ laboratory, have written protocols for all the different kinds of DNA tests that it performs?
Yes, we do.
And do you follow those protocols when you perform the tests?
Yes.
Did you follow those written protocols when you performed the tests on the evidence in this case?
Yes, I did.
Are those written protocols based on the scientific literature that you've described that was in existence before the DOJ lab began investigating the D1S80 marker?
Yes, the D1S80 protocol is based on the scientific literature, but that protocol was written specifically for D1S80 in our laboratory, but it compiled the literature from the outside.
Now, in performing the PCR D1S80 test, Mr. Sims has just generally described the sorts of positive and negative controls that are involved in DQ alpha tests. Are there also positive and negative controls when you perform the D1S80 test?
Yes, there are.
And did you listen to Mr. Sims's testimony?
Parts of it.
Parts of it. Can you just briefly describe, let's say, the role of negative controls in the D1S80 testing process?
Yes, by negative controls, there are two types of negative controls. One is the negative amplification control, and that's a sample in which it's used during your setup of the amplification, and either distilled water or a buffer is added to the reaction mix, the cocktail, as we also call it, instead of any DNA. And this should respond negatively. Respond negatively when tested by the D1S80 system. Another type of negative control is an extraction blank, and this is a sample that's taken through the full extraction process, and that's to demonstrate whether there's any contaminant in your reagents. And so that also should respond negatively.
When you say respond negatively, could you explain to the jury what you mean by that?
Yes, by responding negatively, meaning no results are obtained. And you'll see with some of the D1S80 gels, you'll see when I show the D1S80 gels that when you run the sample, if no bands are visible, then that's responding negatively. If there is a response in a negative control, a negative amplification control, or a reagent blank, then that analysis needs to be repeated.
Okay, and could you briefly describe the role and the significance of positive controls in this case?
Yes, a positive control is provided in the D1S80 kit, and this is a control of a known type. The sample is put through the amplification process, and it needs to respond properly for D1S80. And in this particular case, you'll see on some of the gels, it's an 1831, meaning there's two bands that are present, and these two bands need to be present in the proper locations for the samples to be, for the gel to be acceptable.
Could you tell us what you mean by that? Why they have to be in that position for the gel to be acceptable?
Well, it's testing both the amplification and the typing process. And if these bands aren't present, then it shows that there was some problem with your amplification or your typing.
Did you also test substrate controls that were provided to you in this case?
Yes, I did.
And what was the purpose in testing those substrate controls that were provided to you?
Substrate controls are important to show you what the background of the area being tested is. In this case, substrate controls also served as negative controls. There are a few instances where there was some activity in a substrate control. But in general, these all responded negatively for the amplification process at D1S80.
Could you explain what you mean by those few instances?
I believe one of them was addressed by Mr. Sims yesterday or the day before, and that is a control that he had detected blood on, a substrate control on a garment that he had detected blood on. And so that responded with a faint reaction in the D1S80 sample.
And that was from item number 81, Mr. Goldman's shirt.
Yes, it was.
Okay. What other instance were you referring to when you mentioned that?
I believe,
If you need to look at your notes.
yeah, I need to refer to my notes.
Okay.
Yeah, yes, Mr. Goldman's shirt was the only one that had some activity in it.
Okay, and Mr. Sims addressed that when he testified several days ago?
Yes, he did.
Okay. Just generally speaking, could you describe your role in relation to Mr. Sims in this case? What was the official relationship, and how did you get samples from him?
Gary Sims was assigned this case. He was in charge of the case, and he went through the majority of the extractions and documentation of all the samples. I was assigned to this case because of my experience with D1S80. So there are a few samples that I did the extraction and the analysis on, but predominantly Mr. Sims we consulted over how much sample should be amplified, and then I amplified the sample for D1S80 and analyzed it and did the interpretation on that.
Okay, so the closest, or strike that, you never actually physically examined and sampled any item of evidence that was sent to the lab?
No, all the evidence went through Gary Sims.
Okay, and then he would provide you with whatever was agreed upon you needed to do your tests?
Yes.
Okay. I want to ask you if the number items I'm going to ask you if these are all items that you processed and achieved D1S80 results on, and then we'll show some of them, okay? So
Okay.
did you achieve D1S80 results on the following samples in this case? Item number 6 from the Rockingham residence.
Yes.
Item 9G1, 9 is the right-hand glove from Rockingham. Stain G1,
Yes.
G2,
Yes.
G3,
Yes.
G4,
Yes.
G9,
Correct.
G10,
Yes.
G11,
Yes.
G12,
Yes.
G13,
Yes.
G14.
Yes.
Now we'll shift items to item 13, the pair of socks from Mr. Simpson's bedroom. Did you obtain results from stain 13A1?
A results were obtained. That work was done by Steve Myers.
13A2,
Yes.
13A3,
Yes.
13A4.
Yes.
Now shifting to the other sock, 13B1,
Yes.
13B2,
Yes.
and now shifting to the white Bronco, Mr. Simpson's Bronco. Did you obtain results from item 30,
Yes.
31?
Yes.
Staying with the Bronco, 293.
Yes.
From the carpet, 303, 304, and 305 from the passenger side of the console.
Yes.
Okay, and then did you also obtain results, did the laboratory also obtain results from the following items from the Bundy residence? These are drops along the walkway, 47, the one closest to the victims,
Yes.
48 on the Bundy walk,
Yes.
50 on the Bundy walk,
Yes.
52 out in the driveway at Bundy.
Yes.
Did you also obtain results from clothing items from the two victims in this case? Mr. Goldman's jeans, item 79.
Yes.
Mr. Goldman's shirt, which you mentioned a couple of minutes ago, number 81.
Yes.
Number 86, Nicole Brown's dress.
Yes, and that work was done by Steve
by
Myers.
Did you also produce results from 3 items from the nail clippings and scrapings from Nicole Brown, generally item 84?
Yes.
And did you also produce results from Bundy, the 3 stains on the rear gate, 115, 116, and 117?
Yes.
Okay, and again, just to lump these together in categories, some of those results produce mixtures, is that correct?
That's right.
Where you could tell there were mixtures.
Correct.
We'll show some of them in a couple of minutes, but were the mixtures for items 9G1, the glove again, the right-hand glove from Rockingham,
Yes, that was a mixture.
9G2,
That also was a mixture.
9G4,
Yes, that was a mixture.
9G10,
That also was a mixture.
9G11,
Yes, that was a
Not-
mixture.
9G12,
Yes, that was a mixture.
9G13,
A mixture.
9G14.
And also a mixture.
And shifting to Mr. Simpson's white Bronco, did you obtain results that you determined to be mixtures from item 31 on the console?
Yes, that was a mixture.
Item 303 from the passenger side of the console in the white Bronco?
Yes, that was a mixture also.
304 from the same side of the console in the white Bronco?
Yes.
And 305, was that also a mixture from the white Bronco?
Yes, it was.
Okay. Now, Your Honor, at this time, I'd request to have marked a series of films. Ms. Montgomery, what do you call these films, just so we don't confuse the jury with autores? What are they called?
Well, I call them duplicate copies of the D1S80 gels.
Could you describe the final product and how we end up with a piece of film from that product? And what I'd like you to do is just to distinguish between these films and autores, which this jury has seen quite a few of.
Okay. The final product, the similar film is used to get a duplicate of the gel to just show you an original gel that an original gel is placed onto a nylon, actually it's a plastic piece of material, and it's dried down. And then to make copies for court, because we don't want our originals to be submitted to court, we would like to keep them with the files. To make copies of it, we just use duplicating film, and we expose it to light and then put it through a film processing apparatus or machine to get a duplicate copy.
And is that what I have, these films that are produced from the gel that was dried down?
Yes, it is.
Okay, Your Honor, I have a total of 18 of them. I'd like to have them marked as 275A through R. I think that's the right number.
So mark 275A through R.
And I don't intend to show all of these. I intend to show A through H. And I've got them separated out. I'm not sure how. Perhaps I can just describe each one for the record, and then we'll give it the right letter then, Your Honor?
All right.
Okay. And I've shown some of them to counsel. I showed this one yesterday.
These, Mr. Harmon.
Excuse me, right on the Elmo.
On the Elmo?
Yes. Back to you. We're going to use the telestrator a little bit too. So, Ms. Montgomery, why don't you come on down here?
All right, Ms. Montgomery, would you keep your voice up while you're down there, please?
Ms. Montgomery, even though I'm behind you, talk to the jury so they can hear you better than I can, okay?
Okay.
The first copy that I'd like to put on the Elmo is just an illustrative film. Looks like it has nothing to do with the casework in this case. So may that be 275A?
275A.
Okay, Ms. Montgomery.
Could you shift it so it's, so the sample's at the top? Whoops. There we go. And do you think?
Okay, Ms. Montgomery, can we get the jury oriented to what all those markings are and then describe the significance of 275A?
Yes, this is a D1S80 gel, and for orientation purposes, let's see. Ah, there we go. Okay. The origin is here, and this is where it's up at the top. You can't see it on this particular gel, but that's where the samples are loaded, and they're distinct wells, separated, each sample separated. The ones with the multiple banding patterns, these are ladders, and these are made by the company. They're provided within the kit, and they range from a 14 allele down here. Oops, there we go. Hold on. 14 allele or a 14 band, all the way up to a 41. And the way I like to look at D1S80 is the marriage of PCR and RFLP. And by saying the marriage of PCR and RFLP, it's using a PCR-based system, but it's looking at repetitive regions or repetitive sequences of DNA within the genome, much like Dr. Cotton described with the RFLP process. So these are repeat sequences ranging from 14 repeat units all the way up to 41 repeat units. And what the company did is they made some of the alleles bolder, so it's for ease of picking out the alleles. And the ones they made bolder were the 18 allele, which is 18 repeat units, and that's right here, the 24, and then also the 31 and the 34 repeat units.
Now, is it true that everybody matches some allele or some band in that ladder?
Yes, this is termed a discrete allele system, meaning that unlike the RFLP process, with this process, you could use the ladder, the 14 to 41, as a ruler or a reference point, and then just look at your evidence sample or your unknown sample and match up the band to the corresponding ladder length. So, for example, this, let's see. That,
Sample 1, you've just identified.
the sample right here where this is moving, that is considered an 18 allele. If you look at this one,
In sample 2, you put an arrow.
yeah, sample 2, that's a 24 allele or 24 repeat.
Now, are some of these more common than others?
Yes, some of them are. And what the company did, just let me put that down. What the company did is the ones that they made darker are the ones that tend to be more commonly seen in the population. So as you can see on this gel, I'll just make lines. Whoa. Okay, let's not do the line. The 18 allele, the one right here, is seen in 3 of the samples. And that's a total of, 13 samples.
Now, who are those people, samples 1 through 12?
These are just individuals that were taken from our convicted offender database. They're individuals that had been released from prison, and their samples were run. Their DNA was extracted, and they run on the D1S80 system. But then if you also look back to Mr. Harmon's question, if you look at this, the 24, you could see that there are 4 individuals out of the 13. I'm saying 13 individuals because I'm looking at the positive control also. It would be 14 individuals. 4 of these have a 24 allele. But by looking at the second allele, you can differentiate between all 12 of these individuals.
So they share one band or frequently in common, but they're different with the other band quite frequently?
Yes,
Among these 12 people, they're totally different.
correct.
So let me put that another way. From among these randomly selected 12 people, do any of the 12 share the same 2 bands?
I would check to randomly selected. There's no foundation that that occurred.
Yes, of these 12 individuals, none of them are the same type. But it's not unusual for individuals to be. There are instances where individuals are the same type.
Why don't we just start from left to right, just so the jury can get a sense for how you can look at that and describe which type is there, starting with sample 1.
Okay. Sample 1, if we count 14, I guess I should go back to talking about the ladder. It goes from 14 repeats all the way up to 41 repeats, but the 15 repeats missing. So we go from 14, 16, 17, 18, 19, so on. 24, this one's bold again. So sample number 1 would be an 18, 25. Sample number 2 would be a 24, 25. Sample number 3 would be a 24 homozygote, meaning that individual only has a single banding pattern.
Why is that?
What they inherited from their parents was the same allele, 24 allele from both the mother and the father. And then this one, we could continue on with this.
Would you do that?
Okay, this one is a 22, 26. And then this one, sample number 5, is a single banded pattern, a homozygote.
So could you just give us a little better explanation? Why is there just one band there?
Well, there's just one band visualized because both of the bands, the individual is a 24, or not in this case, but this case, the individual is a 28, 28. And so both of the bands are running to the same location. So all you're seeing is a single banded pattern on that individual.
And how can you tell there are really two bands there?
This system's been optimized, so you would see the banding equal intensity of the two alleles that are present, or the two bands that are present on an individual. This is a reference blood stain, and a single band is observed. And in this system, it hasn't been found that any of the samples run off the gel or anything such as that. So if you see a single banded pattern, you can assign that a genotype, or what you're observing and the type of the actual sample.
Okay, why don't you move to sample 6?
Sample 6 would be, once again, I have to get my orientation and count from the 24, 25, 26, 27, 28. So this would be a 28, and this would be a 34. And you could see that 34 band's a little more intense than the other bands. And that, once again, is for ease of, so you can quickly call, or you can readily identify types without having to count from the very bottom up to the top.
Okay, and then sample 7?
Sample 7 is a 20, because 18, 19, 20, and then a 28. And you could see 3 individuals, number 5, 6, and 7, all share the same allele.
But not any other allele.
Correct.
Okay, let's go to sample 8. How would you type that?
Sample number 8 is, once again, here's an 18 allele and 22.
All right, Mr. Harmon, I think we get the point.
Good. Okay, then we can move on. Why don't we move on to 275B, which you assign numbers to each of these gels when you run them, is that right?
Correct.
And let's move to AG148. You recognize that number?
Yes, the number sounds familiar.
Okay, could we put 275B, which we'll identify as gel AG148? Now, what's the purpose of this first gel? Or strike that. Are those labels normally on your gels? The OJ Simpson, R. Goldman, N. Brown labels?
No, those labels were placed on the gel for to identify what sample was in that particular lane.
And why did you just run, is there any evidence run on this gel, 275B?
No.
Why did you run just the 3 reference samples without any evidence on them?
Well, for 2 reasons. First of all, this gel these samples were run prior to any evidence coming into the laboratory. The only evidence that we had obtained were these 3 samples. And as I stated earlier, Gary Sims had examined the reference samples and placed a portion into a tube, and then I extracted these samples and analyzed them for D1S80.
Why did you do that just on the reference samples?
Well, for 2 reasons. First of all, the LA District Attorney's Office asked me to see if we could differentiate between these 3 individuals.
Why was that important?
And that was important to determine whether the evidence was sent to our laboratory for D1S80 analysis. If we couldn't distinguish between these 3 individuals, then it wouldn't offer any more information. At that time, they wanted to know if these 3 individuals could be differentiated.
Okay, could you just briefly describe the layout of this gel? You've already pointed out the ladders. Are there 6 ladders on this gel?
7.
7? There. What else, starting from left to right, inside the left-hand ladder, is in each lane?
Okay, I should have stated on the last gel, what we have a conventional way of setting up these gels. And first is to, after your first ladder, is to place the positive amplification control. And this is once again, the amplification control that's provided in the D1S80 kit. And the amplification control is an 1831. And the next lane on this gel is a negative amplification blank. And this produced no results. And then a ladder, and this is a QC sample, and I believe Mr. Sims went into the explanation of what a QC sample was. And this is run...
Excuse me, does that mean quality control?
Yes, QC stands for quality control sample.
And the next lane over?
And the next lane over is a extraction blank. And this provided no banding pattern.
Okay, and then we've got a lane labeled OJ Simpson
Yes,
after the ladder.
correct.
That's from Mr. Simpson's known reference blood.
Yes, it is.
What type was Mr. Simpson with the D1S80 marker?
He's a 24,25, and you can see there's the 24. Well,
Oh.
24, 25.
And then Mr. Goldman, what type did he produce?
Mr. Goldman is a single-banded pattern, a 24 homozygote.
Okay, that means he inherited the same size band from his mother and his father?
Correct.
Could you move that, or could we maybe rotate it 90 degrees and come up from?
Sure.
Okay, now that's Mr. Goldman, is a 24, so by coincidence, he shares one of these bands with Mr. Simpson.
Correct. And as I stated, the 24 allele is one of the more common alleles seen.
Are there population studies that continue to be produced that compile the frequency of all these bands with the D1S80 system?
Yes.
And I believe you just said 24. Is that the most common allele?
Yeah, it's one of the more common alleles that is seen.
Okay, and when you type Nicole Brown's known blood sample, what type did she produce?
She came out an 18 homozygote, which is one of the other more common alleles that is seen. The 18 and 24 alleles are the most common alleles that are seen in the population.
Okay, now let's just stop here for a second, and I want to ask you, is it necessary to run these reference samples again with all the other tests that you perform on the evidence in this case?
And no, it's not.
Why not?
If you know the results of the individuals, you don't need to run them on each subsequent gel that's run. There's a limitation in the number of samples that can be run on each gel, and that's 20 samples or 20 lanes, and that doesn't include all the controls or the ladders. So to run them once is sufficient for the analysis.
Okay, could we, why don't you move on then to 275C, Your Honor, which .275C, which is AG 168. Do you recognize that number, AG 168?
Yes, the number's familiar.
Your Honor, I'd like to, at the same time I do this, because of the samples that are on there, to have the jury be able to see the photo boards from Bundy and Rockingham. Those are exhibits 120 and 165. So we can see the relationship of these items. Could we do that?
I have no objection to that. May I request that anytime we put arrows on something, we print it out? Apparently that last one didn't get printed out.
Oh, I didn't intend for it, but from now on I will. Sure.
All right, and at the break, we'll reproduce that other one
Sure.
on 275B.
Okay. While Mr. Fairtlow is getting those boards out, essentially you have the same generic layout of the gel. This is a different gel than 148, the one we just saw, is that correct?
Correct.
And I notice you have lanes labeled with Mr. Simpson's name on it, Mr. Goldman's name on it, and Nicole Brown's name on it. Why did you include them on this gel?
They were included on this gel because I had extra room on the gel to run their samples again, and for the analysis. And it's also good, just for the first gel, I wanted to see all the samples placed together.
Okay, and did Mr. Simpson's reference type produce the same type as we just saw in 275B, Exhibit 275B?
Yes, it did.
And did Mr. Goldman's produce the same type as we saw in 275B?
Yes.
And Ms. Brown's, her blood sample also produce the same type as 275C?
Yes, it did.
Okay. Now, we could,
This gel's, some of the samples are fainter, so I'll need to get my glasses.
sure. Can we lower it? There we go. Montgomery, let's start out with the same that was labeled Rockingham number 6. It's displayed on the Rockingham exhibit board, and it's been identified as individually photo 120C. What type did that produce in the V1S80 system?
That's a 24-25, and
Go ahead.
as you can see, there's the 24 allele, and there's the 25 allele.
And that's consistent with Mr. Simpson?
Yes.
And,
Actually, I think it would be clearer to put the arrows this way.
well,
Is that clear?
You're not going to be able to do it across the board, though.
Okay.
But can we get it coming in straight from the top?
Yes. Okay.
Okay, now which allele is that from the top?
That's the 25.
Did you mark that? And then-
Mm.
Back, let's, just to make it easier, why don't we just go all the way across either the top or the bottom? The other band is a 24?
Correct. These bands that I'm pointing to are 24s.
Okay, and the top bands are 25s. Could you mark all of those, please? And
Yep.
if we go over to Mr. Simpson's reference type and show us how they correspond.
Yes, this is the 25. And there's 24 allele.
So you got the same results from the stain in Mr. Simpson's driveway with D1S80 marker as you did with 47, the drop closest to the victims, 48, next down the walkway, and 50, the next one down the walkway.
Correct.
And those results were inconsistent with Mr. Goldman and Ms. Brown.
Assuming a single source, yes, it's inconsistent with Mr. Goldman, and Nicole Brown can be eliminated because her 18 band isn't seen in the bottom region of any of those samples.
Was there any indication of a mixture in any of those stains?
No, there wasn't. By looking at the DQ alpha results also, there was no indication of a mixture on that, on those particular samples.
Okay. Why don't we capture that one? And that'd be,
That'll be equals 275 C1. All right, are we done with the Rockingham exhibit?
yes, we are.
With regards to this, all right, Mr. Farrell, how about if we change places so the jurors can see the Bundy exhibit? Because it's too low with use of the screen.
Yes, sir. I will need to shift these.
Your Honor, I have an objection, and I think we need to approach. All right, let's call order, please.
And Mr. Harmon, 10:30. All right, Madam Reporter, you set? All right, Mr. Harmon?
Can we have the Bronco photo and result board up there now?
I'm sorry, Mr. Harmon, are you going to do the Bundy?
Oh. Item 48, 47, and 50. Change boards now. Mr. Fairboat to the Bronco photo and result board exhibits 172 and 260. And I'd like to have marked as 275D copy of gel AG 174.
275D as in David.
Yes, Your Honor. While we're getting the board set up, could you describe the samples that are on 275D, which is your AG 174?
Yes, these are, once again, the reference samples from the three individuals. OJ Simpson being a 24-25, Ronald Goldman being a 24 homozygote, and Nicole Brown being an 18 homozygote.
Okay, and do we lose a little fidelity when we project these copies up on the screen?
Yes, we do.
Okay, how do you visualize these things when you're looking for them for the first time to interpret them and write the report?
I look at the actual gel after it's been dried down, and I do my interpretation based on, or I write the results that I see at the time on a run sheet, and then also a second reader reads those gels. That could be done at a different or later date, though. These are blue copies, and first of all, the blue copy is represents what the actual gel, what was present on the actual gel. But when you project it up here, you tend to lose. It's not as sharp and it's not as dark. But I think when you see these printouts, you'll be able to see it better.
Okay, and you had the three reference samples run again.
Yes.
They produce the same results?
Yes.
Item number 30, which was taken from the Bronco center console. Could you tell us what results you obtained from that sample?
Yes, a 24 allele and a 25 allele. And you can see it right.
And you're marking them with the pink arrows?
Yes.
Okay.
Well, let's choose.
The red arrows.
Red arrows.
Okay. And what about Bronco 31, the other center console sample that was provided to you? What results or what bands did you see there?
On the D1S80, a 24 band, and also the 25.
Now, what conclusion can you reach on the 24, 25 with respect to the possibility there's a mixture there?
Well, the interpretation had to be based also on the results of the DQ alpha. And by looking at the DQ alpha results, in conjunction with these D1S80 results, it was determined it was a mixture. By just looking at the D1S80 results alone, a mixture can't be determined.
Okay. But if there were a mixture of Mr. Simpson and Mr. Goldman, would it look just like what you see in 31, the console stain?
Yes, neither of them could be eliminated from these samples.
Because they share a 24 allele?
Correct.
Okay. Why don't you move on to Bronco 293, which was from the driver's side carpet. Tell us what results you obtained from that sample.
Okay. On this sample, there's a single banded pattern, and it's an 18.
And who is that consistent with? Could we make that 18 a different color, if you would?
Sure.
Okay. We're now switched over to green, and you're going to put a
Green.
green arrow by the 18 allele.
Yes.
And who is that consistent with?
That's consistent with Nicole Brown.
And is it inconsistent with Mr. Simpson and Mr. Goldman?
Yes, it is.
Could you put a green arrow over there by Nicole Brown? It's a single banded pattern from her reference sample. And 305, could you first describe what's in 305, and then we'll try to mark it?
Okay, 305 is a mixture, and there are 3 bands present: the 24, the 25, and an 18. And
Oh, I'm sorry.
it's slightly difficult to see that 18 from here. But when you see the actual blue copy, you'll be able to visualize it.
Okay, could you put a green arrow down by the 18? And sample 305, the Bronco sample 305. And then let's go back to the red for the 24, 25 bands that you saw.
Okay. And there's the 24. And the 25.
Okay, we marked the 24 and 25. You know, you said you know that's a mixture. How do you know that?
You know it's a mixture because there are more than 2 bands present. There are 3 bands present in that sample: a 24, a 25, and also the 18. And also the relative intensities of the bands. The 18 allele is much weaker than the 24 and 25 alleles.
What's the significance of that?
It shows that the 18 is a minor contribution in that sample.
Could you explain a little bit better what that means?
Yes, it means the amount of the 18 allele is less than the amount of the 24 allele or the 25 allele.
And is there something about the way this whole kit is designed that'll help you, when you see something faint like that, interpret it?
Yeah, it's both the kit and also the in-laboratory evaluation that the Department of Justice did for D1S80. We optimized the system so when you have a neat blood stain, meaning or DNA, a sample from one individual, that you'll get equal intensity of the two bands. So in this case, what you could see is there's not equal intensity of those bands, and there's a weaker component down there at the 18.
And are the results that you produced for 305 based on D1S80 alone, are they consistent with a mixture from the 3 reference samples in this case? Mr. Simpson, Mr. Brown, and Mr. Goldman and Ms. Brown?
Yes, none of those individuals can be excluded as a possible source of the DNA in the Bronco stain, number 305.
And the DQ alpha results also contribute more information to that mixture, is that true?
Yes,
And that they're still consistent with that mixture?
correct.
All right, tell you what, let's take our break 5 minutes early because we'll have to do another set of exhibits here. Ladies and gentlemen, please remember all of my admonitions to you. Don't discuss this case amongst yourselves. Don't form any opinions about the case. Don't conduct any deliberations till the matter has been submitted to you. Also, do not allow anybody to communicate with you with regard to the case, and we'll take a break for about 15 minutes. All right? Stand in recess. Record should reflect that we've now been rejoined by all the members of our jury panel. All parties are again present. All right, Mr. Harmon, you may continue with your direct examination. All right, let's proceed.
At this time, I'd like to have marked as 275 for identification, gel number AG244.
All right, 275E.
And display for the jury 262 and 260A, which are respectively the stock results board and the stock photo board. Okay, now, Mr. Montgomery, could you please describe, skipping over the positive and negative controls, could you please describe the samples which are of interest to us in this case?
Yes, these are two samples from the sock, two different locations. And you could see them. The first one, described as B1, is a single-banded 18. And the second one, described as B2, is a single-banded pattern also, and that's an 18-18.
And that's consistent with Nicole Brown?
Yes, it is.
And it's inconsistent with the defendant, Mr. Simpson?
Yes,
Inconsistent with Mr. Goldman?
yes.
Now, we're just showing some of the films in this case for the jury's edification, is that correct?
Right.
And the result board actually lists other D1S80 results which were produced in this case. Are those in this package of films that we have as well?
Yes, they are.
Okay. Okay. We move on to 275F, which is gel AG265. Do we have the glove result and photo board, which are equal to 272, A and B?
All right, we'll mark the printout from 275E as 275E1.
Montgomery, while we're getting the board set up, could you please describe the samples that were on this gel that produced the copy of the film which we've labeled 275F?
Yes, this is DNA extracted from 4 different locations on the glove. And G11 G11 is a mixture, and this is,
How can you tell it's a mixture?
there are, you need to see the original, or the copy that's going to be printed out. From here, it's a little difficult to see the minor contributions. But what you could see is the major banding of a 24, and so that's a major type of a 24, 24. And then you can also see a weaker 25 allele, and on this one there's also a weaker 18 allele.
Could we just use some, let's say with 18, could we use a different color? Or let's sort these out by color. The 18 allele, could you put an arrow to the left of it? And then we'll change colors and go up to 24 and then 25. So you put it, could you get it real close there? So you've got a green arrow next to the stain 9G11. That represents the 18 allele.
Yes.
Now, from the 3 reference samples, whom is that consistent with?
And that's consistent with Nicole Brown. She can't be eliminated as a contributor of that weaker 18 allele.
Now, how do you know that's a mixture?
You can tell it's a mixture for 2 reasons. First of all, because there are 3 bands present in this sample. And secondly, because of the relative intensity of the banding pattern. As I stated, that 24 is more intense than either the 18 or the 25. In this sample, it appears the 18 and 25 are relatively equal intensity to each other, but yet that 24 is much stronger.
So what does that mean?
That means that there's more than one individual that could have contributed to that sample.
Okay, and from among the 3 individuals, could you mark the 24 and 25 alleles using different colored arrows, please, to the left? So you're putting a red arrow to the left of
Would you like a different?
the 24 allele, and could you use a different color for the 25 allele? Now, considering the 3 reference samples that were provided to you in this case that you didn't run on this gel, what can you say about the whether or not those three reference types could be contained in the stain at 9G11?
And none of the individuals, none of the 3 reference samples, OJ Simpson, Nicole Brown, or Ronald Goldman, could be excluded as a source of this sample.
And let me ask you that same question about those 3 people, and what's the significance about the weakness of the bands with respect to the 24 allele? Can you say anything more about how these things line up in terms of intensity?
Well, you also can, D1S80 was, or I mean DQ alpha was done on these samples also, and by using both the DQ alpha results and the D1S80 results, you can sort this out a little further.
I don't think DQ alpha was done on this sample, correct?
Oh, I'm sorry, you're right. DQ alpha was not done on the sample.
So can you say anything more about the relative intensities in terms of the combinations of the 3 reference samples in this case?
I can say who could be the major contributor of the sample and who could be the minor contributor. And I can say who of those 3 individuals, none of them could be eliminated as a source of this blood stain.
What could be the type of the major contributor in this case based on your observations on this stain?
By looking at lane G11, glove 9, lane G11, the major contributor is a 24 homozygote. And then the minor components are an 18 allele and a 25 allele.
And from among the 3 reference types, is any one of them a 24 homozygote?
Yes,
Who's that?
Ronald Goldman.
And is the 25 allele unique to one of the 3 reference samples in this case?
Yes.
Who is that?
24, 25 is the type of OJ Simpson.
So from among those 3 people, Nicole Brown, Ronald Goldman, and Mr. Simpson's the only one that has a 25 allele.
Of those 3 individuals, that's correct.
Okay. And the population frequency data actually describes how many other people might have the 25 allele.
That's correct.
Okay, let's move on to G12. Could you describe, before you put any arrows on it, describe the bands that you're able to see on the original and the blue copy that appear in 9G12, if you will.
Yes, G12 is also a mixture, and from here, I'm having a difficult time seeing that the minor contributor, but the minor type, when you look at the gel, you'll see an 18, a band in the 18 region. So in this sample, the major contributor is a 24 homozygote, and the minor contributor has an 18 allele present.
And are those 2 types consistent with the 2 victims in this case, Mr. Goldman and Ms. Brown?
Yes, they are.
When you say major contributor, minor contributor, are you saying that it's a mixture of those 2 types?
By major and minor, I'm able to say what the major type is. As far as the minor component, what I can say is an 18 allele is the minor component, but that 18 can be an 18 homozygote or it could be an 1824.
So there could be just a 1-banded 18 pattern, which would be consistent with Nicole Brown,
Correct.
or there might be another 24 band that's hidden under the 24 band. Is that what you're saying?
That's right, or it could be an 1824.
Okay, can we try to get some arrows in that, let's keep green with the 18, if you will?
Well,
We'll get that green.
great.
And then keep the red with the 24 allele. Okay, let's move over to G13. Will you describe what you see there, and then we'll discuss it?
Yes, G13 is similar to G11. The interpretation is the same on that sample. Where it is a mixture, there is more than one band present. There's a major type and there are minor alleles. The major type is a 24-24, and the minor alleles are an 18 and a 25. And once again, you need to see the printed out copy.
Okay, now, let's look back on G11. Are the results that you've described for G13, are they essentially the same results as you've seen in G11?
Yes.
Are they different in any way?
The 25 and the 18 are slightly weaker in G13, but in general, the same pattern is seen in G13 as G11.
And would you strike that? So the comments that you made about G11 would apply to G13 as well?
Correct.
Could we, using the same colors that we used in G11, could we try to get some arrows in there to show where those bands are? Okay, you've marked with a red arrow the 24 allele.
Yeah, this is getting a bit messy.
You've got the pink arrow on the 25 allele in G13. And then you've marked the 18 allele with the green arrow.
Right.
And once again, in this sample, from among the 3 reference types that were provided to you, Mr. Simpson's the only one that has that 25 allele. Is that correct?
That's correct.
Okay, let's move over to G14, if you will, and describe what you observed in G14.
G14 G14 has a major contribution of a 24 homozygote. And in the 18 region, there's what I call a possible 18. There wasn't a clear, distinct band in that region, and so I wasn't confident to call it a band.
Can you show us where that is with an arrow? Is there something that's more readily apparent when you actually look at the original and look at the copy?
Well, from here, I can't see anything in that region. When you look at the original gel or the copy of the gel, what you could see is a darkening in that region. But as I stated, it's not clearly defined, and I would not call it definitely an 18 band being present.
Okay, and what do you see there in that lane for G14?
There's a 24 allele present. So the major source of the sample is the 24 homozygote.
And I believe Mr. Sims described, because of that possible trace 18, that one could not exclude Nicole Brown.
If that is a band, then no, Nicole Brown could not be eliminated. But as I was saying, I don't feel confident calling it a band. So if it is a band, then she can't be excluded. But since I won't call that a definite band, then that's...
So from among the 3 reference types, who would you exclude as...
I don't think she was finished.
Yes.
Oh.
So that was... So this one, I'm able to say what the major type is on the sample, but as far as a minor contributor, I'm unable to say anything about it.
Okay, because you're not sure there is one.
Exactly.
Okay. So let's look at what you do see there. Who is not excluded as the source of the stain in G14?
I could not eliminate Ronald Goldman as being a possible source since he is a 24 homozygote, and the pattern that's seen in G14 is 24-24.
Okay. Could we capture that, Your Honor, and mark that 275 as 1?
Yes.
And I'd like to move on. I'm going to display the nail board, Your Honor, with those photos at the bottom, the nail kit and the other photos.
All right.
As well as the Bundy result board. The nail board is People's Exhibit 220, and the Bundy result board is 259. time, I'd like to have marked as 275G for identification, gel AG293.
Yeah, Mr. Farrell, let me see 220 before you get it up there, just briefly.
You want to see those photos? That's the one.
I just want to make sure my recollection is, yeah. The, Mr. Bancroft, would you avoid 220, please?
Okay, while Mr. Farrell was getting the board set up, could you describe the samples of interest that were provided to you for D1S80 typing in this case?
Yes, these were extracted samples from the fingernail scrapings of Nicole Simpson, Nicole Brown.
Scrapings and clippings?
Yes.
And as soon as we get the board up there, we can relate the items, because it doesn't seem like we have enough room there. So we slide the, just identify parentheses and describe the items so the jury can correlate them. What is your 45B number, which is labeled fingernail 45, fingernail 84, and then in parentheses 45B?
The 45A is the right fingernail clippings. 1B refers to nearby blood on the same nail.
Okay. And would you describe, starting from left to right, 45B, what results did you obtain with the D1S80 marker when you typed your number 45B?
Those are all single-banded patterns of 18 homozygotes.
That's consistent with Nicole Brown.
Yes, it is.
Could you use, I think we've been using green for the 18 allele. Could you put an arrow for each of the three samples that gave you the same result, the 18 allele? Okay, is there any question in your mind that they're the same result for all three of those samples?
No.
Okay. Then we capture that, Your Honor, it was 275G1.
Yes.
And the last board and results, if we keep the Bundy result board up there and put the Bundy photo board, which is People's Exhibit 165 up there. At the same time, I'd like to have marked as equals 275H for identification, gel AG295. I'd like to talk about is items 115, 116, and 117, so as not to confuse them with photo ID numbers, which are in the upper left-hand corner and then just down below it, the items from the rear gate at Bundy.
Okay.
And you performed D1S80 typing on those samples as well?
Yes, I did.
And would you please describe your results, and then I'll ask you to mark them if you will.
Yes, these all came back 24, 25. And as you can see,
We're using red for 24, 25. Why don't you mark 115? Mark 275H on the record.
Yes, we have 275H. It's AG 295.
Thank you. So that's a 2425.
yes, it is.
Okay, and would you move over to 116 and describe the results that you obtained on 116?
On 116, it's also a 24, 25.
Okay, we put arrows that show where those bands are. Okay, and on the 117, the rear gate stain, would you describe the results and put arrows to show where they are?
Yes, the same results were obtained with sample 117, and that's a 24 allele and a 25.
Are those results, or strike that, is 115 consistent with Mr. Simpson's D1S80 type?
Yes, it is.
Is 116 consistent with Mr. Simpson's D1S80 type?
Yes, it is.
117 consistent with Mr. Simpson's D1S80 type?
Yes, it is.
Are those three types, the three results, 115, 116, 117, are they consistent with one another?
Yes, they are.
Are all three of those results inconsistent with having come from Ronald Goldman alone?
Well, yes, because by looking at the D1S80 results and also by looking at the DQ alpha results, there's no indication that a mixture is present here. And so on this, the type of the individual would be a 24, 25.
And is Nicole Brown excluded as a source of those stains?
Yes, there was no 18 allele detected on any of these stains.
All right, could we capture that as 275H1?
Yes.
Now, did anything happen in the course of your testing on all the samples that you've tested in this case that undermines your confidence in the results that both you and Mr. Sims have been presenting?
No.
And as I mentioned, there are a number of other films that have not been shown to the jury.
That's correct.
Thanks, Ms. Montgomery. I have no further questions, Your Honor.
All right. Ladies and gentlemen, we're going to take a brief recess to recycle the exhibits and allow Mr. Blazer some opportunity to look at some items. I'll ask you just to step back into the jury room and we'll buzz you out in probably about 10 or 15 minutes. All right? Thank you.
Now that they forced us to try to put things on our result boards, why don't we put who they could be from then? I think that would be fair and consistent, Your Honor, to put what it is, not what it's not, because then you
And
get...
what do you suggest?
Well, that's where it gets a little convoluted, because there are all sorts of mixtures or single stains. I can go down the list, but that's the point. To say what it's not doesn't mean anything. To say what it is provides the proper context for it. I can go down the list if you'd like.
Well, it's your record, counsel.
Okay, well, I think it's argumentative to say that there are no matches or however they want to describe it, because that ignores what it is. They can clearly say that in argument, but this is not the point to start preaching your argument.
Well, let me ask a question. Is this dealing just with the clothing or
Just the clothing, yes.
just the clothing?
We can change that to OJS excluded. I mean, this is from their test results.
Well, I thought this wasn't the time for argument in your exhibits. I mean, that's what the court has consistently ruled
Yes.
when we've had to modify almost every one of our boards, and a statement is a statement. An argument about what it's not is an argument.
their results.
He's excluded from all the stains on the clothing. That's all it says.
Do you agree with that?
Did
With
he-
regards to the clothing, not the glove, the clothing.
Sure, but you know, how you say it, I mean, we have results. The results say who they could be from. I'm assuming this is in the context of the series of slides, it's the last slide that, it is the summary of the sum total of stains that were tested, but it's still argumentative. That's in fact the final outcome of this process. But this.
All right. Mr. Blazier, I'm going to direct you to add summary results, Ray clothing, victim's clothing, that the term carryover be changed to mixtures, plural. And you can put defendant excluded.
I can.
Yeah. All right. With regards to the D1S80 results, Rockingham glove, Mr. Harmon, do you have any objections to this?
It's going to be worded. They're
The-
supposed to point out where the stains are. So since this is in a sequence of slides, it's hard to appreciate what it's supposed to demonstrate with
All right.
what.
My concern, Mr. Blazier, is with the second glove slide, where you have no genotypes consistent with OJS. And that does not appear to be correct from the evidence that's here.
I think the only stain that has alleles consistent with Mr. Simpson are in the wrist area. Those 3, G, 10, 11, and 13. They're mixtures of consistent with Mr. Goldman and Nicole Brown Simpson in the body of the glove, but nothing consistent with Mr. Simpson.
Is the idea that you're trying to convey here is that the wrist area is the only area where you have something consistent with, and then for the remainder of the glove, it's not consistent?
Correct. Then all the tests done on the rest of the glove, he's excluded.
All right, any other comment?
Yeah, it. It'd be nice if we had overnight to look at these, so we don't have to struggle with them now, Your Honor.
Cross-examination.
I know it's cross-examination, but he had them yesterday, and we could avoid this stuff, so. You know, this says it all. This is a photograph of the gloves. One of them's on the inside, and the others are on the outside, and this just has a circle around the cuff of the glove, Your Honor.
As simple as I could make it. Every time I put more stuff on there, I'd get objected to.
May be simple, but it's not accurate. Sometimes they
How is it
deviate,
inaccurate?
you know.
How is it inaccurate?
All right, Mr. Harris, can you make these corrections?
You want to write it down?
All right.
On the last slide, rather than defendant excluded, can we say OJS excluded?
Yes.
See, I mean, it's just one side of the glove, Your Honor. There's two sides to it.
I understand. All right, any other comment? Mr. Harmon?
Just that it oversimplifies something and it's misleading. I mean, we have actual photographs of it that actually depict where they are, and to force somebody to say, yeah, that's right, when it's not, I don't think we do anybody a service with that.
All right, with the corrections ordered by the court, the other objections will be overruled.
Thank you, Victor.
All right, let's have the jurors, please. I'm talking to Mr. Blazer.
We had 2 additional films that should be included in the,
Hold on just a second.
sorry.
All right. Good morning again, Ms. Montgomery. You're reminded you're still under oath. And Mr. Harmon, I'm sorry, I thought you had completed your direct examination.
Well, we noticed there were 2 additional films, so we should go up to T then, 275T.
All right, 275T. Mrs. Robinson, do you have that? All right, thank you. All right, good morning, Mr. Blazer.
Good morning, Your Honor.
All right, you may commence your cross-examination.
Ms. Montgomery, good morning.
Good morning, Mr.
Folks,
Blazer.
good morning. Ms. Montgomery, when Mr. Harmon was asking you questions about various stains, he was describing the locations where those stains were purportedly obtained from. Do you remember that?
Yes.
Now, you have no personal knowledge of that those descriptions were accurate, do you?
No, as I stated, Mr. Sims looked at the samples that came into the lab, and then I did the analysis for D1S80.
And of course, he doesn't have any personal knowledge either, does he? He was not out at the scene collecting evidence.
No, he was not.
And nobody from the Department of Justice, to your knowledge, was out there actually collecting and preserving evidence, correct?
Correct.
And so you have no way of personally verifying that the evidence was collected properly or processed properly before it came to your lab. Is that correct?
I was not at the crime scene, no.
So you take what is sent to you, and you can account for what happens once you get it, but you can't really say a whole lot about what might have happened to it before.
Correct.
Okay. I want to ask you a couple of questions about your notes. Your notes were provided to us in discovery, were they not?
Yes, they were.
Now, you write your notes in ink,
Yes, I do.
and all of your pages are numbered sequentially, are they not?
Yes, they are.
And you number those as you go along, correct?
Yes, I do.
And when you're done with a block of pages, you go back and actually put page 2 of 70, page 3 of 70, page 4 of 70, do you not?
Well, when I go back, since I number them at the time I'm making the notes, what I do is I go back and I count how many total pages. In this case, there was over 100 pages just for my analysis. And so I'll just write of or slash with the total on the bottom.
And that's so that you can ensure in your own mind that you're accounting for all of your work papers, correct?
Correct.
And anybody else that's looking at your work papers can tell how many pages they're supposed to be, how many pages there are.
Right.
And your handwriting's pretty easy to read, is it not?
I find it easy to read.
When you make a correction, you don't erase anything, you line it out, you put your initials on it, correct?
Right.
And that's all standard laboratory procedures at the Department of Justice, is it not?
Yes, it is.
And those are just good techniques that you've learned as a criminalist, correct?
Well, actually, I learned them prior to being a criminalist, yes.
And in fact, you don't even leave any space at the bottom of a page. You draw a box or an X if you finish something and there's still room on the page, correct?
I like to try to do that. There are occasions when I don't. It's just to let me know that nothing else has been written on that page at that time.
And is it accurate to say that one of the reasons for all of these safeguards that you use is so nobody can come in and change your notes or add something without you knowing about it?
Well, no, I think it's more for, first of all, the numbering is so I know how many pages there are in the order of my notes. And also then when I get into court, so I can easily refer to a certain page and see the sequence of events there. And as far as crossing out, it's just so I'm aware that nothing else was written on that page that day and just ending the page.
Is it accurate that one of the ideas of doing your notes as thoroughly as you do is that at some point in the future when you may get called to court, you want to be able to sit down and reconstruct everything important that you did on a particular case?
Yes.
And furthermore, another important reason for doing it that way is that if for some reason you had changed employment or changed professions or something like that, another analyst could sit down with your notes and basically reconstruct everything you did on a particular test.
Correct.
Would you agree that it's not good procedure for you if you get called into court to have to say you don't remember something because you didn't write it down?
Well, it depends on what that is.
Any important steps that you do in a particular test, if you say, I don't remember because I didn't write it down, that's not very effective on your part, is it?
Sustained.
Mr. Montgomery, I want to ask you just a few questions about your background. Mr. Harmon asked you about undergraduate courses that related to DNA, and I didn't think you said any. Was I accurate in that?
Yes, I think I jumped right into post-graduation courses. I didn't discuss the courses that I took as an undergrad, but some of those courses were biochemistry, in which DNA was discussed. Also, in my environmental toxicology courses, DNA was discussed in several of those courses.
In what context?
In the context of like mode of action, how chemicals reacted on humans or animals, and so it was discussed on how the DNA was affected by certain chemicals in the environment or that were produced by chemical companies.
Did anything in your undergraduate background cover forensic applications of DNA technology?
No.
Now, you indicated in... Now, did any of your graduate courses involve forensic applications of DNA technology?
Well, as far as graduate courses, there was only, well, actually, two of the courses I took post-graduation, I received graduate units for. The others were under or upper division courses, but not graduate courses. And the ones that I obtained graduate units were the FBI Academy class through the University of Virginia, and that was 6 units of graduate course, and that was the forensic application of DNA technology, and also the laboratory course. And then also 1 unit of graduate level units was given in the DNA sequencing course through the University of Northern Colorado.
Now, your genetics course in 1992 at Cal State Hayward, that didn't deal with forensic applications of DNA technology, did it?
No.
How about your 2 semesters in molecular biology at Berkeley, 92 and 93? Did that deal with forensic applications of DNA technology at all?
I'm not quite sure if it went over. The instructor of the course had an interest in forensics and the application of DNA technology to forensics, but I don't recall if he actually had a lecture on it or not.
Now, that particular course, I think you indicated that there was no hands-on work in that course.
That's correct.
Your training at Quantico, did that involve any study or applications or casework dealing with D1S80?
I believe they talked about AMFLIPs, which the amplified fragment length polymorphisms, in the PCR section, but the course was not devoted to D1S80 analysis. But they did talk about the PCR technique.
But specifically, it relates to D1S80. That wasn't really a focus of that course, was it?
It was not the focus of the course.
Did you have any hands-on work at all at the Quantico course?
Yes, I had hands-on.
Any involving D1S80?
No.
Any involving DQ alpha?
Yes.
And how much hands-on work did you have there?
Half of the course was devoted to the laboratory aspect of DNA analysis, and the other half was devoted to lectures. So approximately 2 weeks, maybe a little less than 2 weeks in total of that 4 weeks was devoted to the laboratory aspect of DNA analysis.
And approximately how many individual tests did you do at that time? Just approximately.
Oh, I don't recall.
More than 3?
No, I can't imagine it was more than 3.
Now, your statistics course in the fall of '93 at Berkeley, did that have anything to do with the forensic applications of DNA technology?
No, but that instructor was also interested in the application of forensics and how statistics was applied to forensic issues. And so he would discuss in passing just some of the forensic applications and how statistics related to that.
And how about your course in DNA sequencing in the summer of '94 at the University of Colorado? Was that dealing with forensic applications of DNA technology?
Well, the instructor of that course was Dr. Steve Lee, and he's a researcher at our laboratory. And so he did discuss forensic applications. But the focus of the class was DNA sequencing, just in general.
Is it fair to say that, that all of your background, with the exception of the Quantico training course and what you've talked about with the California Criminalistics Institute, didn't really focus on forensic applications of DNA technology to any great extent?
My classroom, my formal classroom training, is that?
In these courses that we've talked about at Colorado, at Berkeley.
Yes, as far as the formal classroom training, the credited, or I mean, I guess I should say credited, not formal, those involved just the theory. But then the courses that I took that weren't credited, such as the course through the California Criminalistics Institute on PCR, that was devoted to DNA analysis and its forensic aspects.
Now, you listed some of the courses that you took under general criminalistics, and I wrote those down as basic microscopy, zone electrophoresis, sexual assault, low explosives, clandestine labs, arson investigations, and basic serology. Is that a pretty complete list?
Yes, I believe that's the extent.
Would you agree that all of those, with the exception of perhaps zone electrophoresis, deal with things other than forensic applications of DNA technology?
Correct.
And your 1989 course in crime scene investigations that you took in Eureka, did that have anything to do with DNA technology?
No, it was a crime scene investigation course, so reconstruction and collection of evidence.
I'm sorry.
I'm done.
And your firearm safety course in '89, that didn't have anything to do with DNA, did it?
No.
Have you written any, you have any publications that deal with DNA technology?
No, I don't.
Now, when you went to work at Modesto in 1988, that was an arm of the Department of Justice?
Yes, the Modesto is a satellite laboratory. It's a full-service criminalistics laboratory.
And you were there for 3 years?
Right.
Did you do anything relating to forensic applications of DNA technology during that 3 years?
No, not hands-on. At that time, I was interested in DNA, the use of DNA in forensics, and so I would keep up to date on what was going on in the DNA field.
But you had no practical experience at all?
No.
Now, when you went to Stockton in 1991, again, you were doing things other than DNA, forensic applications of DNA technology, correct?
Correct.
So that didn't provide you with any relevant experience to what you're doing now? Relatives.
Well, as far as DNA analysis, that's correct.
Okay. Now, finally, in 1992, you moved to the DNA lab in Berkeley.
Right.
When you moved there, did you go there with the intention of working in forensic DNA technology areas?
Yes, I did. The field was changing from conventional serology to DNA analysis. And since I was very interested in DNA analysis from early on, I decided the best place to gain more experience and to actually work with the technology was at the Berkeley laboratory.
And at what point did you get assigned to, you were put in charge of setting up the D1S80 program?
Two individuals, myself and Richard Showalter, were to work together in the development of a system, the D1S80 system.
And when was that about, approximately?
That was, I believe Richard started it in June, the summer of '94. So I think he began around June with the technique. And then when I got back from the FBI Academy, I worked with him, and we worked together in the development of that system.
When you were assigned that responsibility, had you ever done a D1S80 test?
Done a,
D1S80 test?
no, I had not.
Perhaps this might be a good time.
All right, ladies and gentlemen, we're going to take our break for the lunch recess. Please remember all my admonitions to you. Don't discuss the case amongst yourselves. Don't form any opinions about the case. Don't conduct any deliberations till the matter has been submitted to you. Do not allow anybody to communicate with you with regard to the case. We'll stand in recess until 1 p.m. All right, Ms. Montgomery, you may step down. Your order to return 1 p.m.
Where there's no CDOT and they're inconclusive, and I wanted to make a time with the court as to maybe at the end of the day or whenever we could take up these matters.
I have the feeling we're going to finish with Ms. Montgomery today.
Yes, we should, and that's why I was hoping that we could take up those other matters, and
Right.
perhaps we can finish all these witnesses this week.
Right. Good. All right, let's have the jurors, please. The members of our jury panel, good afternoon, ladies and gentlemen. All right, Ms. Montgomery, would you resume the witness stand, please? All right, good afternoon, Ms. Montgomery.
Good afternoon.
You're reminded, ma'am, you are still under oath, and Mr. Blazier, you may continue with your cross-examination.
Thank you, Your Honor. Good afternoon, folks. Ms. Montgomery?
Mr. White?
When we broke for lunch, we were talking about your level of experience with the D1S80 system. When the program started at DOJ, was there anyone at DOJ that had any experience at all with D1S80?
Yes, Dr. Nora Rudin had looked at some of the gel systems, I believe it was back in 1990, and that was for a brief period. And at that time, our lab decided to focus on other things. And so the project was dropped. Dr. Nora Rudin was a researcher at our laboratory.
And that's the extent of the experience of everybody in your lab with D1S80 prior to when you started, when you were in charge of the program setting it up?
Correct.
Now, the period of time from when you started to set up this program until it was actually implemented in casework was 8 months?
Yes, it was. We started in, well, actually, it was a little over 8 months. We started in, like I said, around June of 94, and then began casework in April, or June of 93, and then began casework in April of 94.
Is there any requirement at all that a new marker system such as D1S80 or a new technique have any kind of government approval at all to assure that it's a valid system?
Government approval?
Yes.
No, but there are guidelines that are set up that must be met, and some of these guidelines are, well, actually, these guidelines are addressed under TWIG-DAM, and TWIG-DAM's the Technical Working Group on DNA Analysis and Methods, and they set up criteria that must be addressed.
Excuse me, Ms. Montgomery, the answer to the question was regarding government.
Okay.
There is no government approval process required at all, is there?
No, there's not.
And you're aware that with clinical testing involving new DNA techniques, they require extensive governmental regulation and approval by the FDA.
Actions irrelevant in such application.
Sustain.
But there's no such approval process like that required for a new system such as D1S80.
No such approval is made.
Sustain.
You indicated TWIG-DAM that there are guidelines that you must follow. TWIG-DAM is a voluntary program, isn't it?
I believe, well, in our laboratory, it's not a voluntary program. It's something that must be followed. The guidelines have to be followed, first of all, for accreditation purposes, and also because our laboratory believes in TWIG-DAM guidelines.
There's no enforcement mechanism, however, is there, if you don't follow the guidelines? Are you aware of any?
I'm not aware of any.
And TWIG-DAM is a voluntary, you can run your lab without complying with TWIG-DAM, can't you?
I'm not quite sure.
Oh,
So you're not sure whether TWIG-DAM is mandatory or not, in terms of you being governed by someone other than yourselves?
No, I'm not.
Now, the D1S80 system is really the newest system compared to DQ alpha, polymarkers, and RFLP, is it not?
Well, compared to those three, yes, but there are other techniques that are being looked at this time.
There are other techniques that are even newer than D1S80, aren't there?
I'm sorry, not techniques, I mean markers. Because the technique in general is the PCR technique, but it's actually the marker that's the new part of it.
Well, but this is, you described it as a marriage between RFLP and PCR. It's a different system than just PCR and just RFLP, isn't it?
Correct, it's incorporating the two techniques into one.
And just as it might have the strengths of both systems, it also has the weaknesses of both systems put together.
Protection of foundations, facts.
Sustain.
Any indication that it works any better than PCR DQ alpha or any better than RFLP?
Well, I believe that all three of these systems are equally reliable and can give reproducible results. I don't see any of them as being better than the other as far as obtaining results. The difference is, as Dr. Cotton and Mr. Sims stated, is with the RFLP that you can you need larger amounts of samples and et cetera.
You're aware of the areas of PCR technology where there can be problems such as contamination, are you not?
Yes, we're always concerned with contamination.
And that applies with D1S80, does it not?
Yes, it does.
Because it is a PCR process.
Correct.
So it has all of the potential problems that DQ alpha testing has, does it not?
Correct. As far as sensitivity and contamination, yes.
Now, with respect to the RFLP half of it, does it have some of the limitations that RFLP has with respect to the size of a sample that you need?
No, it doesn't. And I guess I need to clarify how I meant by incorporating both RFLP and PCR. By incorporating the RFLP, by the marriage of it, we're looking at these repetitive sequences. And so just to clarify that issue, that's how it's similar to the RFLP process. As far as your question, I'm sorry, could you restate your question?
I think you answered it. Now, as being responsible for setting up this program, you're required or responsible for reviewing all of the relevant literature pertaining to D1S80, are you not?
Yes, I reviewed the literature.
And did you spend a considerable amount of time doing that to make sure that you understood the technology?
Yes.
And do you still keep up with that?
Yes, I do.
Would you agree with that with respect to D1S80, there's much, much less literature on it than there is, for instance, RFLP?
Yes, that's true.
And there's much, much less literature than there is with PCR DQ alpha, correct?
That's correct.
It has been used in casework much less than PCR DQ alpha and RFLP, correct?
Yes, DQ alpha's been around since 1986 or so.
And during the course of your work at DOJ, you've only worked on 20 cases, I believe. Is that what you said, approximately?
Yes, approximately 20 cases. Each case involves more than just one sample, though.
And how many cases does your lab approximately in a year?
Counsel, you want to, we're talking DNA cases?
DNA cases.
Well, our laboratory is not known for quick turnaround time. We tend to be a bit slow at doing cases. So each analyst does approximately 2 cases a month, and that's what we strive for. And in our lab currently, we only have 4.5 individuals doing cases. So in the past year, this would just be a guess, but let's say probably around
100?
80 to 100 cases.
And so that in the 20 cases that you've worked on, is that all of the D1S80 cases?
No, it's not.
Would it be fair to say that the D1S80 cases are a relatively small percentage of the total cases that DOJ handles with DNA testing?
Well, anytime there's a small amount of sample, both the DQ alpha and the D1S80 system would be used, unless the individual is excluded on one of the systems, then the other system wouldn't be used at that time. So anytime we have the capability of using both systems will be used. If it's an RFLP case, then typically we only do the RFLP and we don't do the PCR typing.
Now, Cellmark doesn't do D1S80, do they?
No, they don't.
As of August of last year, had Department of Justice reported or testified about any D1S80 results in any case anywhere?
Yes.
How many? Approximately.
Well, I know of one case, and that's a case that I did in, I believe it was the summer last year, in '94. I don't recall if any other cases were, if any of the other analysts have testified to D1S80.
So all you can recall, I'm sorry.
So at this time, all I can recall is the one case that I've done.
Now, you were asked some questions about proficiency testing, and I think you indicated that you had been involved in 5 proficiency tests,
Yes.
all D1S80?
Actually, if I could review a document, I
Sure.
can answer that for you. And I believe this is a document that you've obtained on discovery.
And how many of those proficiency tests involve D1S80?
4 of the 5 involved D1S80.
And you indicated that you got those all right.
Correct.
And what does that mean? Does that mean that you got the type that was expected that you get?
Yes, that means that the results that were reported out by the laboratory were the expected results.
Now, are these blind tests? Do you know you're being tested?
Yes, we know we're being tested.
And who prepares the samples?
Prepares them to send out to our laboratory?
No, yeah. Are those prepared in-house?
No, these proficiencies are prepared externally. We subscribe to 2 systems. One is CTS and another one is Cellmark. And we also have in-house proficiencies that have to be done too.
But these 5, or the 4 that related to D1S80, were they externally provided or internally provided?
Yes, they were externally provided.
But you knew you were being tested.
Correct.
Did you know what the results were supposed to be?
No, I didn't.
Let me show you.
All right, with the code portal.
Yes, I do.
And those are, that's just one proficiency test, is it not? It was rehybridized twice or a second time?
Yes, this is one proficiency test.
So you're testing the same samples in both of those worksheets.
Correct.
but you're doing a different hybridization. Those represent two different hybridizations.
Yes, and there are some different samples.
Okay. And I'd like to have these marked.
Mrs. Robertson? All right, 1172.
1172 A and B, or?
Yeah, A and B.
I'd like to put 1172A on the Elmo first. Chris? Ms. Montgomery, the two pages I showed you, one was numbered page 6 of 9, and the other was page 8 of 9. You recall that?
I'd have to look at it again.
Vince, I want you to tell me which one of these you did first.
Well, actually, that's not my writing at the top of the page. My writing's at the bottom, and I can't tell the whole. It hasn't been Xeroxed completely.
Can you tell from your own notes?
Well, this is a proficiency. This isn't something that I brought to court with me today.
Okay.
But it appears that one says 14 of 22, and the other says 19 of, and there's a 2. So, if I could see the original documents or a better copy, that would be even better. But just by looking at it, it appears that the 14 of 22, the other one's trying to say 19 of 22, maybe.
Let me show you the rest of the documents that those pages came from, see if that helps you.
Yeah, it appears that the second page is probably part of the same document. It has the same proficiency number, and it also has a date 2 days later. So this is the first one that has the date of the 18th of October, and the second one would be the 20th of October.
All right, for the record, she's referring to 1172A is the first one, and 1172B is the second one.
Thank you.
Well, let's look at 1172A. Let me give you the rest of this packet in case you need to refer to it.
And do you also have the original or a copy of the photos that go with these?
Now, this is the worksheet. What we have up in the Elmo is the worksheet that describes what results you saw after you performed the test, correct?
You know, this multimedia, I can't decide what to look at.
Whatever's easier.
You know, the clarity's not very good on this, so maybe if I came down there, it would be easy for me to see.
Sure.
Okay.
Can we pull it in a little bit, because it's 432-51, right there, that's good. 432-51, that line refers to one of the samples that was provided in the proficiency test, correct?
Actually, that's dash S. That's an S.
Okay. That's one of the samples you were supposed to type and get a result, correct?
Correct.
And QC 839, in the middle of the screen there, toward the bottom, what's that?
That's a quality control. What would be more helpful is if I had the whole file on this. It's difficult just to take this all out of context to actually see what the notes are prior to the samples being written on here. Because a lot of times what you want to do, when you talk about sample, you can't write everything that pertains to that particular sample. So you write a certain section in that sample. And then in your the body of your notes, you write a little more description.
What you write in the little boxes there is what you see on the dots. Or,
I'm sorry,
I'm sorry.
I'm talking about the sample lane.
Okay. Now, this is actually a DQ alpha proficiency test, correct?
Correct.
And what you write in the boxes there is what you see on the dots, correct?
Correct. Now you're over to the dots.
Yeah. On QC 839, do you see where it says there's a hint of a 1.3?
Yes, I see that.
That's not the correct result for QC 839, is it?
No, that must be the correct result because this goes through approval. We have this approval process where, first of all, a second reader reads the strips, and then the results are given to a supervisor to review the whole case. And the QCs are unknown to us. And the supervisor reviews the results of your QC. And if you and they check to make sure those QCs are giving the proper results. If the QC doesn't give the proper results, then something needs to be done.
The box under type, that's the type of that quality control sample, is it not?
Correct.
And that's indicated as a 1.1, comma 3, correct?
Right.
Nothing in there about a 1.3, is there?
Oh no, because that's a hint. It's
What's a hint?
a hint is just an indication of some. I believe Gary Sims talked to you yesterday or the day about, or a couple days ago about this. When you look at the DQ alpha strips where there are a series of the dots, and next to the 1.3, in this case, if I wrote that there's a hint, then there was some outline or a hint of some activity seen at that dot, but it was significantly less than the C dot. So it was, it had to be documented, but it wasn't significant to the analysis. When in our laboratories, things need to be C or greater before they're actually called.
If this were a mixture, however, you interpret mixtures where you have dots less than the C dot as being alleles, don't you?
Dots less than the C dot?
Yes.
If you see a C dot, then you do report that out. As far as a C minus or a trace or a hint, you note it, but as far as any interpretation, one needs to be cautious about interpretation on that.
You're not supposed to see anything at the 1.3 dot with a quality control sample that's a 1.13, are you?
It's argumentative. Stay. Rephrase the question.
Are you supposed to see anything in the 1.3 dot with a quality control sample that it's a 1.1 and a 3?
Well, if you shouldn't see a substantial amount of activity at that 1.3 if that is a 1.1, comma 3. It's one of the limitations of the system. At the 1.3, you could get cross hybridization, and this is where a sequence that's complementary or to the 1.3 will actually cause some hybrid or cause some of the 1.3 dot to show up. But this is just an artifact of the system, and at such a low level, one wouldn't even interpret that result.
So you can get 1.3 dots when there's nothing there.
When there's no,
What? When there's, let me rephrase it. You can get a 1.3 dot when there's no DNA there?
it's not a dot that would be interpretable. As I stated, that's a hint. It's not a C dot. The intensity is substantially less than that C dot.
What's the difference between a hint and a trace?
We have in our laboratory, we have a scale of progression on how to call these dots. An outline hint means there's you could see something on the strip, but it's just a small amount. A trace is a little more, and then there's a C minus, and then a C, and then a C plus.
What's a trace? Is a trace indication of some DNA there?
A trace is a little more than a hint, but once again, it's not significant for the analysis. It just happens to be an artifact or a limitation of the system, or not a limitation, an artifact of the system. A trace would not be interpreted in the results.
What's the difference between a system that gives you artifacts and a system that has limitations that gives you an artifact?
Action, that's argumentative compound.
World.
As you indicated, it wasn't a limitation of the system.
Right.
Whose fault is it that you get dots at 1.3 when there's no DNA there?
That's argumentative.
Sustain. Phrase it quick.
Are the 1.3 dots caused by limitations in the system or something else?
Well, it's caused by a little of both. And what happens, these are not significant dots. They're hints and traces. They would not be counted in your interpretation. But what happens is with slight variations in the typing, some trace amounts of 1.3 activity can be seen on the strips. And I guess one could say it's a limitation, as Mr. Blazier says, but also it's an artifact of the system. And if it was something, these dots are so faint and so weak that, as I have said, it just wouldn't be counted in your interpretation.
It could also be a minor component of a mixture that has a 1.3 in a small quantity in it, couldn't it?
It could be, but it wouldn't be interpreted at that level.
And is there some sort of uniform scale within the forensic community by which you determine the difference between a hint and a trace?
No.
Does it have anything to do with the results that you expect to get out of a test as to whether you're going to call it a hint or a trace or a C minus?
Actually, that's argumentative.
World.
No.
Now, the positive control, the lane second from the bottom, that was supposed to be a 1.14, correct? That's what the type that was put in that lane, correct?
That is a 1.1, 4.
Now, you got a trace of a 1.3 in that lane, correct?
Correct.
And that's more than the hint in the quality control sample.
Yes, it is. As I said on our scale of progression, a trace is slightly greater than a hint.
Now, are you supposed to get these kinds of traces and hints when you perform tests? Are they expected?
No, they're not.
So this is an unexpected result, is it not?
This is something that it's not unexpected. I think unexpected is a bad word to use with it. It's something, as I said, it's just an artifact of the system, and by doing reanalysis, you could probably get rid of those hints and traces.
Okay, let's talk about the reanalysis, which is the page 2, correct?
Correct.
This is 1172B. Now, you did this one again, the one we just looked at, and the sheet that's on the Elmo is the results of the second run or second hybridization on the same samples. Is that correct?
Yes, some of the samples are the same.
Why did you run it the second time?
I'd have to see the notes to know why.
Is there anything in that packet there that tells you?
No.
Ordinarily, what would be your reason for running a proficiency test a second time?
There could be various reasons. I think I'd need to see my actual notes to be able to explain why I reran this the second time.
Would one reason be the hints and the traces that you got the first time?
Actually, positive speculation.
Sustain.
If the results that you got on the first run were totally as expected, would you have any reason to run it a second time?
Actually, positive speculation.
World.
Well, That's possible. You know, I would need to see the actual, to speculate on this, or it's best not to speculate. It's best to actually see my notes in this particular instance.
Okay, now these are your notes with respect to how you read the strips, correct?
Right.
Now let's look at 432S1, the top lane. That's the first one. Now, would you agree that you found that there was a hint at 1.3 and a hint at 1.1 that was unexpected?
Once again, by saying unexpected, I think you're putting more emphasis on it than you should. I did notice hints in both of those samples, and once again, the hint is not significant to the results of that analysis.
So these are hints that show up now on the same sample that didn't show up the first time you did it, correct?
Could you put the first one back up?
You've got the first one in your hand.
Oh, you're right. Yes. Yes.
They did not show up the first time, did they?
Correct.
Now in 433S2, we've got a trace at 1.1 and a trace at 1.3 that don't conform to the genotype of the sample, which is 1.2, 3, correct?
Correct.
And that didn't show up the first time you did that sample, did it?
That's correct.
434S3, we now have a trace at 1.1 that is not consistent with the type of that sample, which is a 4, correct?
Right.
Now, quality control sample 839, we have 2 hints that are inconsistent with the genotype of that quality control, correct?
Well, no, they're not inconsistent with the genotype of that quality control. I have to keep emphasizing that a hint is not significant. A hint is just noting that something, you're seeing a little darkening in the dot, and it's not significant to your interpretation. The results of that QC are 1.1, 3, and those were the correct results.
Isn't one of the tests of whether a system like this is a reliable system, whether or not, if you do the same samples 2 times, you're supposed to get the same results, aren't you?
Yes, and as you can see in this case, the same results were obtained.
Okay, you can resume your seat. So is it fair to say that this one particular proficiency test that we've looked at is an example of one which has been scored in some fashion that you got the right answers?
Yes, the right results were obtained from that proficiency.
Who grades these?
Excuse me?
Who grades these?
Grades them. Individuals review them. First of all, a supervisor in the laboratory would review the results, and then secondly, the outside agency that submitted the samples to us would review our results and then send out a result whether we obtained the correct results or not.
Do you know what a laminar flow hood is?
Yes, I do.
And do you use that performing any kind of PCR amplification, the various steps that you perform in that test, do you do them within a laminar flow hood?
Yes, I do the setup of the amplification in a laminar flow hood.
Would you ever use a hood that is called a flume hood, I believe, that sucks air from the outside of the lab into the area of the hood and then out?
Sustain.
Laminar flow hood, is that something that's pretty standard in terms of your education as a criminalist, that you're taught what that is and how to use it?
Oh, well.
Well, it's just a hood that has airflow in it, a window of air that separates the inside environment from the outside environment. And there's really not much training on it. You just turn the switch on to activate the hood.
But you're taught what a laminar flow hood is, aren't you?
I don't recall being taught. It's just something that I'm aware of, something that I'm familiar with.
The kits that you use to perform DQ alpha and D1S80 tests require that you do them in a laminar flow hood, don't they?
Sustain first question.
The kits that you use that are provided by Roche that produces the kits state that various steps should be done in a laminar flow hood.
Oh,
I'm not quite sure. I don't recall seeing that in there. If you could show me the section.
Look at that. Have any of the proficiency tests that you've taken involved mixed bloodstains?
No, I don't believe so.
Most of the, or many of the stains that you looked at in this case, your test results indicated could be mixed stains, correct?
Some of them, yes.
And so you've never undergone any kind of testing to see whether you're able to do mixed blood stains accurately?
No, that's incorrect.
What kind of testing have you undergone for that?
I've a part of the validation process of the D1S80 marker, I looked at mixed samples, and these were both mixed, they were mixed DNAs. So not actually the mixed blood stain together, but the DNA, or an extraction process where the two DNAs were mixed together. And I looked at that during the validation process in use in our laboratory. And I believe that's the only, those are the mixed bloods that were examined. There are mixed samples that are examined also in the laboratory, and this sexual assault samples, vaginal swabs where you'll have epithelial cells, and then if there's sperm present also.
But there are no proficiency tests that you've taken that involve mixed samples of blood, are there?
Well, I was just looking at this as you were, or as I was talking, and it's one of the proficiencies I did was a blood semen mix. And that was a cell mark proficiency. And so that's where some of the blood and semen was mixed together. It wasn't two blood stains, but it was two different samples mixed together, and one of them happened to be a blood stain.
Well, without getting into any detail, sexual assault samples are different than mixed blood samples, aren't they?
Right, but this, correct.
You have other methods that you can use, a differential lysis, to split them apart, can't you?
Correct,
Which you can't do with blood stains.
right.
Incidentally, did you perform any kind of analysis on any of the stains that you looked at under the microscope to determine whether you could tell if there was a saliva component to any of these mixed stains?
No, as I stated earlier, Mr. Sims looked at all the evidence initially, and I did the D1S80 or DQ alpha analysis on the samples after he examined the samples.
So you have no way of determining whether any of the components of the mixtures you've testified to have saliva contributions from a source as opposed to blood?
Correct.
Now, I want to ask you some questions about the victim's clothing with respect to the D1S80 results. Do you have that in mind?
Yes, I'll pull up the notes.
Slide. We need to have a number for slide presentation.
all right, next in order will be 1173. 1173.
And we have the fourth slide, which is slide D.
Mrs. Robertson, 1173.
Now, could we have slide E? Now, Ms. Montgomery, you conducted tests on 3 items of clothing, correct?
I conducted analysis on 2 items of clothing, and Steve Myers in our laboratory examined Nicole Simpson's dress.
Okay. And the 2 items that you looked at were the shirt and the pants of Ronald Goldman.
Correct.
And the dress was looked at by Steve Myers. That was Nicole Brown Simpson's dress.
Actually, the DNA was analyzed. The items were looked at by someone else.
Okay. And there were certain D1S80 results that came out of that analysis of those 3 items, correct?
Correct.
Now, I've indicated on my chart up there that I just color-coded it so it's a little easier to understand. We have slide F. Now, would you agree that from your results, yours and Steve Meyer's results, there were 6 stains looked at on Nicole Brown Simpson's dress, 3 of which indicated a contribution by, possibly by, Ronald Goldman?
I'm referring to the report at
Sure.
the time. Yes, a total of 6 stains were examined, and 3 of the stains had a weaker contribution of a 24 allele.
Now, can we have slide G, please? Would you agree that there were 9 stains examined from Ronald Goldman's genes, 5 of which had an indication of an 18 allele, which is consistent with Nicole Brown Simpson?
Well, 4 of them had a weaker 18 allele. One of them had G6 from the left thigh area, had a possible trace 18, and that's what I had talked about earlier, where there was a darkening on the band, but it could not definitely be determined that it was a band.
Okay. And we have slide H. Would you agree that there were 8 stains examined from Ronald Goldman's shirt, and all 8 of those stains showed an 18 allele consistent with Nicole Brown Simpson?
That's correct.
Now, can we have slide I, please? To summarize, there were a total of 23 stains tested on these 3 items of clothing, correct?
You added up the numbers that you...
Yes.
Okay, yes. I'll take your word for it.
And, I'm sorry, 16 of those stains, let's make that 15 since you said the one was just a trace, indicated mixtures containing possibly both victims.
Correct.
Now, is it also accurate to say that of all of those 23 stains, OJ Simpson is excluded?
That's correct.
Now, would you agree that, can we go back to slide H, please? It's H. Would you agree that this is a fairly, withdraw. The shirt that was tested, these were stains that were taken from all over the shirt, correct?
I'll have to look at the notes.
Let me withdraw that one, I'll ask you another one. Did you do any of the cuttings on the shirt for these stains?
No, I did not.
And did you examine the shirt at all,
No.
ever? Did you see Gary Sims examining it at all?
No, I don't recall seeing it.
There was a considerable amount of blood on the shirt, was there not? Do you know that?
From some of the photos I've seen, it appeared to be.
Now, would you agree that one way that there could be such carryover from one victim to the other would be if the clothes came into contact in some way?
Actually, also speculation, argument, it's a thing.
Ms. Montgomery, can you tell us, in your opinion, how would you get the kind of mixed stains on both sets of clothing in so many different stains?
Objections are permitted to make all prospective person the way to phrase.
Sustain.
Now, one of the stains on, I believe it was the shirt, was the substrate control, was it not? Actually, it wasn't the stain, it was a control.
Correct, there was a substrate control submitted, or a substrate control taken with that.
And do you know who made the cutting for the substrate control?
No, I do not.
Now, the substrate control, we've had a lot of testimony about what they are, but just very briefly, that's supposed to be an area of the shirt where there's no apparent stain, correct?
All right, it's best to take a substrate control that close to the stain in question, but without any biological fluid that's detectable, such as no blood present.
And would you agree that that substrate control showed the presence of alleles?
Yes, I believe it did.
And is that an indication that there can be enough blood or biological material in a sample to cause a result where you can't see it on the original sample?
Objection assumes facts, not an evidence. That's the case.
Overruled.
Could you rephrase that?
Yeah, there are two possible ways that could happen. One is the controls failed, correct? If you get a reading on a substrate control that's supposed to be empty.
Well, the substrate control is to test what the background of that substrate is.
Okay.
So I wouldn't say it failed.
Well, yeah, failed was probably the wrong term. If it shows something, it indicates that there's DNA there.
Correct.
And this particular control showed that there was DNA in an area of the shirt where there didn't appear to be any stain.
Well, it showed an 18 allele in a region that was called a control.
Was that a yes?
Well, if it was taken as a control, then the individual wanted to see what the background of that material was. And it did show some activity in the background of that material.
But by definition, that would have been an area that didn't have any apparent stain.
Objection is argumentative.
Sustain.
Substrate control, is that supposed to be an area that doesn't show any apparent stain?
It's the same objection.
Overruled.
Yes, as I stated, the substrate control should be an area that's near the stain, but one that does not give you a presumptive a test for blood or anything such as that.
Now, I want to ask you some questions about the specific testing that you performed in this case. Do you have your notes with you?
Yes, I do. Excuse me.
Could you refer to page 5 of your
Okay.
notes?
Mr. Robert.
Doug again.
I'm sorry, what page was that?
5? Now, you, on, I believe it was August 6th of 1994, you started testing and you started to test the reference samples to see what the types were, correct?
Thank you. I believe the analysis began on August 4th of 1994.
And you amplified, or you tried to amplify Mr. Simpson's reference sample the first time, and you got no results, did you?
That's correct.
And that was a sample that you assumed came from Mr. Simpson because it was in the reference file labeled Mr. Simpson, correct?
Oh, actually, that was a tube. Gary Sims examined the blood stains that were submitted, pertaining to OJ Simpson, Ronald Goldman, and Nicole Brown. And he took a portion of the blood stain and put it into a centrifuge tube. And then he gave me those tubes for the extraction process. And so the tube was labeled, I have it specifically, he labeled it with our case number, he labeled it with the evidence number, and then he labeled it OJ Simpson.
Now, that was a sample that you expected to have bands show up, but nothing showed up, correct?
Yes, that's a sample, a reference blood stain from the individual.
Now, you had to do that one over again, didn't you?
Yes.
And that was because you got no bands the first time.
Yes, there was inhibition in that sample, so there was no visible bands on the D1S80 gel for OJ Simpson's reference blood stain.
Now, inhibition is a term that I think we've heard it a couple of times, but that means that something happens with the DNA that the alleles don't amplify. Is that an accurate description?
Yes, it's during the amplification process. Somehow there's inhibition of the polymerase for the amplification of that specific allele.
And that can occur in samples other than reference samples too, can't it?
Oh, yes.
So in mixed samples, you can have inhibition and some of the alleles that might be there don't show up.
That's unlikely. When you have inhibition, the whole sample is inhibited. You don't just have inhibition of select alleles within an amplification.
I thought you just said a question or so ago that you could have alleles that don't show up because of inhibition.
Sustain.
So you're saying that if there's going to be any inhibition at all of the alleles are going to be inhibited?
Yes, you would have inhibition of the sample, not inhibition of one particular allele, but inhibition of the alleles in general.
Can you have partial inhibition in question mark?
I have not seen partial inhibition, no.
You can have differences in the efficiency of the amplification, can you not?
Well, the system has been optimized so you don't get any of this, what was the word you stated?
Inhibition?
Well, the system has been optimized so you don't have some of the partial amplification of alleles.
When you say it's been optimized, what do you mean?
It's been optimized both at the manufacturer and also through validation studies and in-house evaluation of the system. And this, as I was talking about, the twig dam requirements, this was one of the requirements that had to be addressed before D1S80 or any PCR marker can be used in the laboratory.
You're not aware of the phenomenon where a minor component of a mixture might get lost in the amplification process and not show up in typing because of the relative quantities of the two contributors?
Oh, now you're talking a different thing. Now what you're referring to is a stoichiometric effect, where if you have a minor component, that it's possible, depending on how much of that minor component's present, it's possible not to see the other contribution, but that would be at very low levels of DNA.
Would you spell stoichiometric for the report?
STO, I believe I need to write it down.
STOCHIOMETRIC,
Correct.
I think.
We'll accept that.
The test in this case, some of the minor components of the mixtures involved very small amounts of DNA, didn't they?
Small relative to the initial DNA, but not small as I was referring to just in your previous question.
So the amounts that you used in this case, you would never expect to see an allele drop out in amplification?
Well, there were some samples where we amplified a small amount by DQ alpha, and I believe it was, I'd have to look at my notes, but I believe it was 400 picograms that was amplified. But as with D1S80, the smallest amount that was amplified, I believe, was 800, and typically we were amplifying well over 1 nanogram of DNA.
Now, you also had a problem with one of your standards in the D1S80 test that you performed in this case, did you not? I refer you to page 28 and 29 of your notes.
Yes, that's not, that does not have anything to do with the D1S80 system, though. And what that is a slot blot.
Well, the slot blot, you used the slot blot to determine the quantity that you were then going to use for the D1S80 gels, correct?
That's correct.
And one of the standards didn't work right, did it?
Right. On this one of the higher standards, it was a 40 nanogram that was outside of the range that we were even comparing our samples to. These are the standards that are used for comparison with our unknown DNAs. And one of the 40 nanograms was out of balance compared to some of the other standards that were on the gel.
And you had to discard that particular standard because of that, did you not?
Yes, I threw that standard away.
Did you do the slot blot over again?
No, I don't believe I did because the 40 nanogram standard was irrelevant on the results of the other samples that were on the gel because everything was at a lower level and it wasn't, the 40 nanogram standard wasn't used as a comparison for the unknown samples.
So when something goes wrong with the test, you make some kind of an assessment that whether it really would affect your results or not in deciding whether you do something over again?
Yes, one needs to, if something such as that, where the 40 nanogram standard was less intense than the 20 nanogram standard, if something like that happens, one should troubleshoot it and determine what was happening. And if it appears that it had any bearing on, or any bearing on your results, then your analysis needs to be repeated. In this particular case, it was shown that the 40 nanogram standard was irrelevant because what I was looking at was low levels of DNA in the unknown samples. And so I was comparing at the lower levels, the lower level standards as opposed to the 40 nanogram standard. So basically that 40 nanogram standard did not even need to be placed on that gel.
So your answer is yes. Can we put that down as a yes?
You'd have to re-ask the question, please.
You also had a problem. I'll pass on that. You also had a problem with the Rockingham glove, didn't you?
Sustain.
Refer you to page 46 of your notes. First time you did it, you had a problem, didn't you?
Oh yes, that gel was reanalyzed.
And what happened was the ladder was smeared and distorted, and you had some crossover from one lane to another on the gel, correct?
Yes, and this demonstrates that when something's unacceptable in our laboratory, we do reanalysis of the samples. If there's ever any question, then we reanalyze samples.
Now, let me refer you to page 62 of your notes. One of the standards that's used on these gels, and let me ask if we could have some photographs marked. Actually, let me use one of the prosecution's exhibit 275H. Show that on the Elmo, please. I just want to use this for illustrative purposes. We count three lanes over from the left.
I'll need to come down there to.
Okay.
Okay.
3 lanes over from the left is called the 1 nanogram control lane, is it not?
Yes, it is.
And that is a known sample that has genotype 1831, correct?
Yes, that's an additional standard that's placed on the gel. It's provided within the Roche kit. And as you can see, the first lane, if I had the little telestrator, I can
Well,
point to
I'll
it.
show you another picture. We can highlight the other picture.
Okay.
But that is supposed to be, that's supposed to come up 1831 each time that you do it, correct?
Well, that's an additional control. The control that must,
Excuse me, Ms. Montgomery, question was, is that what comes up?
I'm sorry, what?
That 1 nanogram control, that's one of the controls that you use, and that's supposed to light up at 18 and 31 if your test is being done properly.
Yes and no. That is an additional control, but the control in question that must respond properly is the first one in that after the ladder, and that's the 4 nanogram control of an 1831.
And the bands on the 1 nanogram control, should they be of equal intensity?
Yes, yes, they should, but the one in question is actually the 4 nanogram.
No, the one I'm asking about is the 1 nanogram.
Right.
Should those be of equal intensity?
One would want them to be of equal intensity, yes.
Well, you can resume your seat, please. I believe you indicated on direct that your system has been optimized so that bands with equal amounts of DNA will show up with equal intensity. Did I hear that right?
You're correct.
Ms. Montgomery, I showed you a series of pictures, or I gave them to you at lunch for you to look at to see if they were accurate pictures of some of the films that we've already seen, correct?
Correct.
And did you agree that the packet I gave you was an accurate, these were accurate pictures of your films?
Yes, they were.
Your Honor, I need to have these marked. Let me ask you just to look at these again, just to satisfy yourself that they're the same.
All right, Mr. Blazier, I think these would be 1174. How many do you have?
There are 7 pictures. We have photo 96. And this is, Your Honor, what was the first number?
1174.
Now, Ms. Montgomery, this is just a picture of the film that you showed us that contains the Bronco samples number 30, 31, and 293, correct?
Yes.
And I'm going to highlight, or I'm going to magnify the 1 nanogram control lane.
Mr. Belanger, are we going to go through all of these at this time? Would it be easier if I just stayed down at the,
Well, I think you should be able to see these on the monitor. Now, you agree that the lane there on the right is the 1 nanogram control lane?
yes.
Now, you can refer to page 70 of your notes. Would you agree that when you read this, you read the 18 band of that 1 nanogram control as being brighter than the 31 band? It's a little difficult to see in the picture, that's why I referred you to your notes.
You said page 70 of my notes?
Yes.
Could you show me the full scale of this?
The picture? Sure.
Yeah, it's difficult looking at it out of context. Yes, and that's not page 70 of my notes.
Okay. Do you have the page of your notes that shows your worksheet for items 30, 31, and 293? My copy.
Actually, yeah, it says page 44.
Page 44.
Yes.
Would you agree that your notes indicate that the 18 is more intense than the 31?
No, I disagree with that.
Okay.
My notes don't indicate that at all.
All right, take a look at page 70 of your notes. Take that one down. You looking at your notes?
Yes, I am.
That was, that's your run sheet from which samples?
Page 70, this is AG, Analytical Gel 267. And this is a composite of many samples, quality control samples, positive controls from different amplification days, and also two controls two control samples relating to the Bronco, and those are DNA 17. DNA is our DOJ number. 17, which is what, LAPD 30, and then also DNA 18 control, which is LAPD 31.
So that's is that a different run of the same samples that we showed in the last picture?
Well, the first picture you showed was the actual samples that were taken, the samples that related to the controls of the Bronco. They were samples, DNA 17 and DNA 18. Those are two Bronco samples. And then on the next gel, the one you're referring to now, AG 267, those are the controls that go with the Bronco samples on that previous gel.
Okay, and those controls were done at a different time from the samples to which they belonged.
Yes, there's only limited space on a gel. You can't fit, sometimes you can't fit all the samples.
Well, in many of these runs, you did put the control samples with the samples to which they belonged, did you not?
Yes, when there's enough space, I'll put as many samples as I can on the gel. But if there's not enough space, then you're limited with the number you can put on the gel, and you'll have to put it on a separate gel.
Now, would you agree on your run for the controls, page 70 of your notes, that you determined that the 18 band in your standard 1 nanogram standard line was greater than the 31 band?
Whoops.
Arvin, you want to give us a hand?
Dropping notes. Thank
Is that
you.
all?
We don't want any missing pages. No, the positive control that goes with those control samples function properly.
My question was, did you note in your run sheet that 18 was brighter than 31?
Which control?
1 nanogram controlling.
That's fine.
Oh, okay, the 1 nanogram control. Now, this pertains to a different amplification. This doesn't pertain to the controls that were put with the Bronco. And yeah, there was a weak 18, and the 31, there was a hint at the 31. There was a difference in the intensity of those alleles.
And your 4 nanogram control lane also had a difference in intensity, did they not? Did it not?
Yes, it did. There was a gel problem on that region of the gel. That was the farther region of the gel, the last 4 lanes.
That's not the way those controls are supposed to work, is it? They're supposed to be of equal intensity, aren't they?
Yes, we've optimized the amplification so you get equal intensity of those alleles. This was a gel
Somehow this
effect.
one didn't get optimized.
No, this was a gel effect. This isn't an amplification effect.
So there are some effects on the gel that can cause there to be different intensities of bands in the same lane.
Yes, I mean, that was demonstrated on that particular sample, on those samples towards the further region of the gel, that there was a discrepancy between those alleles. And it's a gel polymerization problem.
Now, you also indicated on your worksheet for those controls that your 31 allele was smeared and not well defined. Is that another manifestation of the gel problem?
I'm sorry, which sample?
The same one we were looking at, page 70.
Yes, and that gave you an indication that that region of the gel wasn't functioning properly.
Now, let me refer you to page 62 of your notes. And that was the run for the positive controls for what? Actually, those are SOC samples, are they not?
Yes, they are.
And you have both some controls and some of the evidence samples on that run, correct?
Correct.
And you had a problem on that run as well, did you not, with your 31 band?
No, well, that wasn't a problem because on this run, 4 nanograms. No, that wasn't a problem.
It didn't come out the way it's supposed to come out, did it?
The 1 nanogram control had the 31 band was darker than the 18 band, and that's for the 1 nanogram control. The 4 nanogram control gave equal intensity of the 2 alleles.
The 1 nanogram control is supposed to also give you bands of equal intensity, correct?
Well, the 1 nanogram control, you would like to have equal intensity of those 2 alleles, yes.
And
But
when- Go ahead.
sorry. There are situations where you don't get equal intensity of those 2 alleles. And that's why at lower levels of DNA amplification, you need to be cautious with your interpretation. That's why you strive to amplify more than a nanogram of DNA.
Doesn't that mean that with small concentrations of DNA, you can't really make very precise assessments about band intensity vis-a-vis who the contributor was?
What you're saying with when you have a low level of DNA, that it's difficult to tell which bands go to which bands? Is that what you're saying?
You have to be much more cautious in terms of trying to determine who contributed what bands when you're talking about low levels of DNA.
If you're talking about very low levels of DNA, yes.
Now, we talked about you're being unable to amplify Mr. Simpson's standard, or reference sample the first time, problems with the standard, the glove that you had to redo, the gel problems that you've had, and the 31 control bands that we've talked about. Is this number of problems typical for the D1S80 system?
That's argumentative.
Sustained. Further question.
Do you ordinarily have these types of problems with D1S80, or in this case, did you have more problems than usual?
Sustained.
These kinds of things going wrong, is this typical of this system?
Full vote.
With the amplification, that's a PCR phenomenon, the fact that the reference sample did not amplify. And as far as the other things, the 1 nanogram control is used, or the 1 nanogram standard is used as an additional control. The main control that we're concerned with is the 4 nanogram, and we want to see equal intensity to those 4 nanogram controls. The system, I have all the confidence in this system and with the results of this system. So if that answers your question.
I can just strike all of that as non-responsive.
Full vote.
Was that a yes or a no? Are these the kinds of things that happen routinely with the D1S80 system?
Further question.
The things we've just been talking about, are these typical of the D1S80 system?
They're not typical, no.
So more things, like we've described, happened with respect to this case than you would expect to see in other D1S80 cases.
Sustained. Further question.
Okay, you said it's not typical. Would you expect in D1S80 system on a particular case there to be more problems like this or less?
Full vote.
Over 20 gels were run in this particular case, which is more than most cases. And as far as the problems, there were no significant problems. There was reanalysis on a few of the gels, but nothing was significant to cause problems with this result. There were not more problems seen in this particular case than seen with D1S80 analysis, and they weren't major problems.
So when you said that this was not typical, you were indicating that in other cases you're likely to see more types of things like this?
Sustained. Further question.
Well, you said this wasn't typical, and I asked you, are there more things, problem areas, let's call them, here than in a typical case? And I don't think you answered that. Could you answer that?
Objection. It's not found. It's argumentative. Calls for speculation.
Full vote.
By typical doesn't mean it's not routine that you see some of these phenomenons occurring. That's what I meant by it's not typical.
So in ordinary cases, you don't see these kinds of things happen. Is that what you said?
In ordinary cases. I'm not quite sure what you mean.
We have photo 100, please. And this will be 1175.
1175.
Now, Ms. Montgomery, could you take a look at this and tell me which run this is? This is, I'll tell you, it's AG 184, correct?
Correct.
And this was the run where you did samples from the Bundy drops, correct?
Yes, it was.
Now, on this sample, you did both samples of those drops as well as some of the controls, did you not?
Correct.
And the writing at the top is, that just identifies what's in the lane below it, correct?
Yes, that's actually, that's Dr. Blake's writing in his photographs.
But do you concur that he's properly labeled the lanes?
Yes, he has.
Now, the last two lanes over here, I've got the little arrow on. The first one is DOJ 24, and what is that?
DOJ 24, as written here, is what we call DNA 24. Dr. Blake tended to call our samples DOJ, and in-house we called them DNA. And that relates to LAPD number 50 control.
Now, that's a substrate control for a Bundy drop, correct?
I believe it is, yes.
And that shouldn't have any lanes in it, should it? Any bands in it?
Correct.
What is that, Ms. Montgomery?
It's not a band. That's, yeah, if you look at the, yes, if you look at the original gel, you can see that there's some blip or glitch. I'm not sure if it's actually cut out of the gel or not. That's right in between the two lanes, but it's not a band. It's definitely not a band.
Would you agree that if that were a band, that means that your control, the substrate control, showed the presence of DNA that shouldn't be there?
Sustained.
Now, how do you tell the difference between something like that that looks like a band and something that is a very faint band?
Well, first of all, this is right in between two lanes. And samples are loaded into the lane, and you would see a distinct banding pattern within that well where they were loaded. As I'm holding up a gel to demonstrate for you, this is where the wells, where samples are loaded. Can you see this? And there's a separation between each where each sample's loaded. That blip right in there is a diffuse, smeary blip in the gel, and it could either be, like I said, it could be just where the gel's been darkened right in that area, and when it was photoed, it was captured by that. But it's definitely not a band. It's not a distinct thickened line.
That's not something you expect to see, is it?
Well, you wouldn't expect to see it, but it's not anything that concerns me.
It indicates that something is going on there that shouldn't be going on there. Isn't that accurate?
No, I think that's inaccurate. Accurate to say that.
Now, we describe this as having a band-like appearance. Would you concur with that? Looks like it could have been a band?
Yeah, it has there's some darkening in that region of the gel.
And it's at it's at a level on the ladder different from any of the people that you've tested in connection with this case.
Correct.
And it's, I believe it's a 15 allele, isn't it? If that is a band, if that band-like appearance is a band, it's a 15.
Oh,
Well, if you're saying it's a band, then that would be in between that 14 repeat and the 16 repeat. So that would be the 15 repeat.
Now, this might be a good time.
All right, ladies and gentlemen, we're going to take our break. Please remember all of my admonitions to you. We'll be in recess for 15 minutes.
Ms. Montgomery, I just want to clarify that the band-like thing down here that we've been talking about corresponds to the lane at the top for the substrate control, does it not? 50C,
Yes,
and for a Bundy drop.
correct.
Take that down.
Well, I'm sorry, actually, you should also include that extraction blank in the second, or in the lane next to the ladder, because since it's right in between those two lanes, you need to address both those lanes.
So it could be either one, correct?
Well, I mean, that little artifact's right in between the two.
Now, the extraction blank shouldn't show anything either, should it?
Correct.
Okay, take it down. Now, I want to ask you a couple of questions about the D1S80 system in general that you talked about on direct. You indicated that it was a discrete allele system, correct?
Correct.
And you indicated that it was everybody matches some of the bands on the ladder.
No, I don't believe I stated it that way.
What'd you state with respect to the ladder encompassing all the possible alleles?
Okay, the ladder is a composite ladder, and that means that from the 14 allele and then the 15 through the 41 allele are present on that. There are alleles that are greater than the 41, and there's the possibility that a 15 allele has been seen, but it's very rare, and so it's not added into the ladder.
So there are alleles in this system that don't show up on this ladder, correct? Because the ladder's too small.
There are some very rare alleles, like I said, greater than 41, and then also the 15 allele that are not on this ladder.
And those are alleles that could be present in someone's DNA that would not show up in this system, correct?
No, that's not correct. They would not have a corresponding ladder allele to call them. If there was something greater than the top 41 allele, you would see a band up there. And I have seen bands greater than 41, and this is very rare. And so anything that's greater than 41 is called greater than 41, and it can't be lined up with any allele ladder. And it happens so infrequently that the company doesn't feel it's necessary to have ladder alleles up in that region, which I agree with.
But the point, you indicated that alleles don't run off the gel. There are alleles in this system that do run off the ladder in the sense that you can't measure them, correct? With the ladder.
Correct, I just stated that, yes.
And now when you say a discrete allele system, is a good analogy there comparing a digital watch with an analog watch that goes around? A discrete allele system is like a digital watch, meaning that you're either at 1 second, 2 seconds, 3 seconds, you can't be in between? Is that what you mean by discrete allele?
You could use that analogy.
And a clock, for instance, is more like an RFLP system where you can have alleles spread out from the top to the bottom of the gel. They're not going to be at discrete positions.
Continuous. Or another analogy would be shoe size. When you go in to buy shoes, they have 8.5, 9, 9.5, but you might be in between that 8.5 and 9. So for RFLP, that's how RFLP is addressed. Whereas with D1S80, each person would fit perfectly into that.
And that would be because that everybody who is a 25 has the same sequence of DNA at that location as anybody else that's a 25.
Well, they have the same number of repeats. So an individual that's a 25 would have 20 repeats, or an individual that's a 25 would have 25 continuous repeats of that particular sequence. There can be subtle variation, and an individual that would be an 18 would have 18 continuous repeats of that region.
Well, when you say there can be subtle variations, what you're saying is that there can be people who show up as a 25, for instance, that have a different sequence as someone else that shows up as a 25, correct?
Oh, yes, that's possible. What we're looking at is length differences. We're not looking at sequence differences with this system.
So two people that come up as a 25 would match in your system, but if they had a different sequence, they are different people, aren't they?
Yes, that's true.
So your system does not have the ability, even though you call it a discrete allele system, to identify precisely the sequence in a particular band.
Right, we're looking at length differences.
And unlike PCR DQ alpha, which does identify specific sequences of DNA, correct?
Correct.
So this, in a sense, is, this aspect of the system is closer to the RFLP system where you're estimating sequences, in a sense, correct?
Well, we're not estimating sequences. We're not looking at the sequence. We're looking at the length differences between individuals, how many repeats they have. It's a 16 base pair repeat. So an individual that's an 18 has 18 of these 16 base pair repeats. And they would have a repeat sequence that's, say, this long, whereas an individual that's a 25 would have repeat sequences that are even longer. The same repeat sequence, the same number of repeats, but just a different length.
The point is, different alleles would come up as the same 25, 24, or whatever, and they'd be different alleles, wouldn't they?
If there is a sequence difference between two individuals, they can run to the same position, but it's the length difference we're looking at, not the sequence difference.
Now, there's also reports in the literature about bands in the D1S80 system not lining up correctly with the ladder, correct?
That's correct.
And there have been reports in the literature of bands going to a place in the ladder that's different from what the true sequence is, correct?
Going to a place in the ladder different than the true.
I'm sorry.
Repeating your question to myself. If there's a sequence difference, it's possible that there could be just a very subtle variation, but it'll still line up with the ladder, the composite ladder allele that's adjacent to it, provided in the Roche kit.
Well, the literature indicates that there are situations where it's so far out of alignment with the ladder that it can be confused with the band above it.
Sustain first question.
Are you aware of the literature that talks about the mobility of bands being such that you can mistype which rung of the ladder you're on because the band is somewhere in the middle?
Yes, that was early on during the development of the system, and what happened is some of the laboratories were trying to use their conventional serology.
I'm going to object as being non-responsive at this point.
Sustain. You state your question, Counsel. Mobility of bands.
I think she answered with the first part of her answer. I'm satisfied with the first part.
All right.
That was an explanation.
No,
Could you read it back? I think you'll see it's an explanation.
Counsel, you can cover that on redirect. Proceed.
Now, this system also has a characteristic that if samples are degraded, you can lose bands, correct?
You could lose your you could get no results with degraded samples, yes.
You can also selectively lose bands, can't you?
At very, very low levels, it's unusual to lose just.
Is that a yes?
Sustain. First question.
You can lose, well, let me rephrase it. The ladder that's in the D1S80 system comprises fragment lengths that go from about, 300 base pairs up to a couple thousand maybe?
It ranges from a little under 400 base pairs up to over 800 base pairs.
And when a sample degrades, it generally starts to break down. The larger fragments get broken up first. There still may be shorter fragments while the degradation process is going on, correct?
Well, it's a random act that's occurring, the degradation of DNA, and that's just cutting at various locations on the genome.
So you're understanding that when degradation occurs, and DNA fragments are cut up. They're cut up randomly in the sense that, well, let me rephrase that. When you start chopping up the DNA, you lose.
All right, let's have it quiet in the courtroom, please.
When you start chopping up the DNA, it gets cut into successively smaller fragments, correct?
Yes, it does.
And as it degrades, if it fully degrades, you may not see any bands at all, correct?
Correct.
And as it degrades further, the bands that are longer, that represent longer sequences, you no longer can see, correct?
That's correct.
So you can lose the upper bands of a genotype, for instance, if somebody is an 1830. The Perkin-Elmer AmpFlip D1S80 kit manual, are you not?
The product insert?
Yes.
Yes, I am.
And that talks about one of the problems of this system being, one of the interpretation problems, that you may lose larger bands with a degraded sample, does it not?
Sustain.
Let me show you, well, let me ask you, are you aware? Let me show you. Are you familiar with that kit insert?
Yes, I am.
And is that what you have in your hands?
And yes, it is.
And do you rely on that in performing the test that you perform?
Yes, this was, third amplification conditions are used in our laboratory for our analysis.
Please look at page 18, section 8.3.1. You ever read that section before?
Yes, I have.
Would you agree that that says that you may lose larger bands if a sample is degraded?
Yeah, it's what it states is that when you have a single banded pattern, that your results should be interpreted with caution. The company has also done some follow-up research in which they've published a paper in May of
I
19...
object. This is not responsive.
That would, all right, proceed.
And wouldn't you agree that the kit insert also tells you that some alleles don't align up with the ladder? Referring to 8.3.2, right below that.
Yes, that's true.
And does that indicate that there are a couple of reasons why that might happen as well? And that is,
Hold on.
are you familiar with the section that I'm talking about, 8.3.2?
Yes.
And that gives several reasons why you might have bands that don't line up the way they're supposed to with the ladder, correct?
Yes, and these are subtle shifts that they're referring to in this section.
Now, the kit also is warranted for amplification of 2.5 nanograms of DNA or more, correct?
Yes, they state in here that they guarantee results with 2.5 nanograms of DNA.
And many of the tests that you ran in this case were on amounts far less than that, correct? Or I should say components of mixtures.
Yes, we amplified some samples down to 1 nanogram.
Now, the D1S80 system is less informative than the RFLP system because it has fewer alleles. Would you agree with that?
Well, we're looking at one region on the D1S80 system, whereas with RFLP, we're looking at various regions, as Dr. Cotton and Mr. Sims discussed.
Now, let me break it down then. You're only looking at one location on the genome,
Correct.
one place in the DNA. With RFLP, with a single probe, you're also looking at one location in the DNA, correct?
Correct.
And with RFLP, however, you can have many different alleles, 60, 70, 80 different alleles for a given probe, can you not?
Sure, yes, you can.
And in the D1S80 system, you have a fewer number of possible alleles, correct?
Correct.
So there's less variation among people in the D1S80 system than in the RFLP system because there's fewer possible alleles that you could find in people, correct?
Yes, by comparing D1S80 to RFLP profiles.
Now, the 24 allele is extremely common, is it not?
Yes, the 24 allele is one of the more common alleles.
In fact, over a third of the Caucasian population will have that, correct? Approximately?
Yes, I believe approximately 30% will have within their genotype a 24 or an 18 allele.
And that makes this system, in this particular case where both Mr. Simpson and Mr. Goldman have 24 alleles, makes it less informative than if they had more rare alleles, correct?
Well, with Mr. Goldman, he's a 24 homozygote. With Mr. Simpson, you need to look at the other allele that goes along with that 24 allele.
My question is, when you have a 24 allele, it's a less informative system than if you have a more rare allele, correct? Because you can't narrow it down as far in terms of the potential contributors.
Correct. If you had two rare alleles, then it would
It'd be better.
more informative. Right.
And the 18 allele, Nicole Brown-Simpson is an 18-18, that is also a very common allele, is it not?
Correct. By very common, as I just stated a few sentences back, that between the 18 and the 24 alleles, you'll see this in one or that allele in approximately 60% of the population, either the 18 or the 24. But then you also look at the other allele that goes with the individual.
In fact, well, if we do that with 18-18, that's actually a fairly common genotype, is it not? 6% of the population has it, Caucasian population?
I'd have to refer to my notes for the exact
Why don't you do that?
frequency. Yes, and 1818 occurs in approximately 6% of the Caucasian population.
So of 100 people, you would expect there to be 6 people that would have that combination, Caucasians.
Approximately 6 out of 100.
And that, because that allele is involved, that's much less informative than if you had a rarer genotype.
Yes, if you had a rarer one, then you'd have less occurrence in the population.
And the fact that Mr. Goldman shares an allele with Mr. Simpson makes it even less informative because there may be some difficulty in assessing who's responsible for the 24 allele. Would you agree with that?
You're talking about mixtures now?
Yes.
And would it be, I'm sorry, could you restate that?
Because there's an allele in common, that makes it more difficult for you to separate components of a mixture because there's one allele that 2 people have in common. Would you agree with that?
Correct.
Let me ask you to refer to page 91 of your notes. You have that in front of
I'm
you?
afraid I'm going to lose all of them.
Okay.
Okay.
Now, do you and Mr. Sims and the other people in your lab use the same standards to decide whether something is a hint versus a trace versus a C minus versus a C?
As it pertains to D1S80?
Yes.
Yes, there's no standard technology as to a hint trace for D1S80. So what happens is when someone's second reading a gel, they call it, and if they call it something different than the first analyst, then you document it in your notes as to what they, if they aren't calling it a hint, then you write what they're actually calling it.
But the term hint and trace mean different things. Correct?
Yes, a hint would be less than a trace.
Okay. And everybody in your lab conforms to that standard?
Typically, yes.
Is that a standard that's set out anywhere in any of the manuals?
No, it's not.
And would you agree that that has a certain subjective element to it? One examiner might call something a hint, another person might call the same thing a trace.
Overrule.
It's possible. When you get down to those lower levels, a hint would be a hint uniformly throughout the laboratory by DNA casework analyst is something that there's a hint there. There's an indication that something's there, but you can't be confident that it's actually a band. A trace, then uniformly through the lab, is there is a band there. It's a very, very weak band.
Now, you, on page 91 of your notes, that's a run where you were looking at what types of samples? It's your DNA 59.
Those are 4 stains from Mr. Goldman's genes.
Now, would you agree that in your lane number 12 on your work notes, you described that as a 24,24 with a hint of an 18?
Correct.
And the second reader scored it the same way, correct?
Yes, and also the supervisor who reviewed it scored it the same also.
What did you report that as in your report? And I ask you to refer to your report of April 6, 1995, page 8 of 11.
That was reported out as a 24 homozygote with a possible trace 18 allele.
What's the difference between a possible trace and a hint?
Well, there's a difference between actually writing your observations on your worksheet and doing your interpretation. Your worksheet's what you actually observed, and then one needs to think about how they're going to word these reports. And I could have written in the report a hint of an 18 allele, but a possible trace, the words sound more effective than a hint of an 18 allele. So the 3 of us that wrote this report, Mr. Sims, Mr. Myers, and myself, discussed it and tried to come up with the correct wording that would adequately describe what we saw. And what we came up with was possible trace. And that's not saying that there's a trace of an 18, that's saying there's possibly a band there, but we aren't confident calling it.
So when you say you want to do it more effectively, in other words, to put forward the prosecution's case?
No, not at all. To reflect what the evidence, is. Doesn't matter if it helps the prosecution or the defense. We're just reporting the science.
What changed between your reading of the actual film and when you wrote the report to cause you to change the way that was described?
Sustain. Rephrase the question.
Did something change in terms of your analysis of this particular test result? Did you read it again or something change?
No, it's to write a hint in a report, it just doesn't, what does a hint mean in a report? In the laboratory, I know what a hint means, and I'm able to hopefully tell you what a hint means. But in a report, for someone just to pick this report up and be able to read it, whether it's the prosecution or the defense, we need to convey what we're trying to get across to them. And by saying a possible trace, that conveys that we're not confident that a band exists there. And to just say a hint of an 18 is, to me, it's it doesn't state it as effectively as saying a possible trace.
Now, unlike RFLP technology, you don't use any kind of computer aids to determine whether there's a band there and whether it's a hint or a trace or a possible trace, do you?
No, that's one of the great things about this system. We get to use the basics.
You just look at it and you say, looks like a trace to me or looks like a hint to this guy, right?
No, it's all based on one's experience also.
And every analyst that looks at these things always comes up with the same terminology, hint, trace, possible trace?
Well,
Oh,
well, I can't, outside of our laboratory, I'm not quite sure if people would put the same words to these. It's in our laboratory, it's pretty well defined. And as you can see on this particular one, 3 different individuals read it and they all came up with the same word, hint, at 18.
Have you ever done any studies where you have looked at different quantities of DNA in a mixture, for instance, to see what happens when you start reducing the quantity to determine at what point a band becomes an artifact?
Yes, I've done studies to demonstrate at which level or at what level you start losing a minor component.
And have you published any studies that show the differences in intensity of the bands between a hint and a trace and a C minus?
No, I have not.
And there's no scale that you use to do this, is there?
No, it's a scale that's made in our laboratory.
I'm sorry?
It's made in our laboratory, and it's just a scale of progression, as I described earlier.
Now, as a sample degrades, have you done any testing to determine what happens when a sample is degrading, in terms of how it shows up, whether it shows up as a band or an artifact?
Yes, I did some temperature studies in the laboratory to see what happened when various samples were subjected to various temperatures for differing amounts of time.
It's not like a light switch, is it, where it's either there or it isn't there. There are gradations of band intensities, are there not?
That's true, yes.
And you must use your subjective judgment to decide whether something is really a band as opposed to an artifact, correct?
Well, when one gets to the lower level where you're not sure if it is a band, then yes, that's correct.
Now, when you read these things, did you know what genotypes the 2 victims and Mr. Simpson had?
sustain.
All the evidence samples that you read, when you did that, did you know the genotypes of Mr. Simpson and the 2 victims?
Yes, I did.
So you knew what results you expected to get, at least as to some of the contributors, correct?
I didn't know what I expected to get, no.
You knew what alleles were common to this case, did you not?
Yes, I did.
And you knew these were evidence samples from this case, did you not?
Yes, I did.
You didn't have any kind of expectation in your mind that perhaps these evidence samples would produce these bands?
Well, no, because that's part of testing the evidence, is to see who can be excluded and who can't be excluded.
You know, are you familiar with the term examiner bias?
Yes, I've heard that.
What does that mean?
You know, I believe I've only heard it in the context of, let me back up. Examiner bias, I think, would be an individual having some bias on their examination.
Have you ever heard it used in the context of someone who knows the results they're supposed to get, subconsciously getting the results that they think they're supposed to get? Not through any trickery or lying or anything, but just you see what you think you're going to see.
So someone could produce bands on the gel? Is that what you're stating?
No, someone might interpret one thing as an artifact or as a faint band, depending on what they expect to see.
Yeah, I'm not aware of that happening, no.
You're not aware of any studies that talk about that particular phenomenon?
I haven't read any studies that address that, no.
Now, the you indicated that the range of size of the fragments in the D1S80 system was about 400 to 800 base pairs,
Roughly, yes.
and the size of the fragments in the DQ alpha system are less than 400 base pairs, correct? It's about 240?
Correct, around 242.
So would you agree that in a degrading sample, you would expect the D1S80 bands to disappear before the DQ alpha allele from a particular piece of DNA?
Yes, that's possible.
So that if you had a mixed sample, let's say in the DQ alpha system, you see evidence of a mixture, but you don't see any extra alleles in the D1S80 system, that might be explained by the fact that the sample is degraded.
Yes, and one needs to also look at their yield gel to determine if degradation was occurring on that, if it's possible.
Well, you're aware that the Bundy drops in this case were severely degraded, or are you aware of that?
Yes, I believe some of the samples were degraded.
Okay. And are you aware of the DQ alpha testing results on the steering wheel sample? That's item 29.
Yes, I am.
You're aware that there was a 4 allele, which is inconsistent with the genotype of any of the 3 people in this case.
Yes, there was a weaker 4 allele detected on the steering wheel sample.
And the reason it was inconsistent because there was no 1.3 to go along with that, which would have been consistent with Ronald Goldman.
All right, this was a very low level amplification, or a low level amplification.
You never tested the steering wheel, item number 29, with the D1S80 system, did you?
No, we didn't.
There's no effort made to try and find out what alleles might be present.
No, there wasn't enough DNA present to analyze it by the D1S80 system.
And how do you know that?
Because I remember discussing that issue with Gary Sims at the time that we were doing the analysis on those samples.
How much DNA was present?
I'll have to refer to Gary Sims' notes to tell you that.
Did that item in your notes?
Yes, I have.
All right, can you give council the page reference?
It's on Gary Sims, page 40. This is another advantage to numbering pages, I guess.
What page?
Gary Sims, 40.
Yes,
36?
40.
Gary Sims, 40.
40.
Sorry.
Compare your notes with Ms. Montgomery's.
Yes, the quantity was approximately 0.9, was it not?
Yes, it was.
Okay.
0.9 what, nanogram?
Nanograms.
Ms. Montgomery? Nanograms?
Correct, 0.9 nanograms. Well, two significant digits, Gary wrote 0.86 nanograms.
And you've performed D1S80 tests on amounts less than that, haven't you?
Yes, I have.
So it wasn't a matter of there not being enough to test, was it?
No, it was a matter of there not being enough to test, due to the fact that this sample was first used for DQ alpha typing, and after using a portion of it for the DQ alpha typing, there wasn't enough sample left for D1S80 typing.
We have photo 95, please. And this will be, what are we up to?
This would be 1176. Robertson, 1176? Yes. Spence, 1176.
Ms. Montgomery, could you look at the monitor and tell me if that appears to be a photograph of your run number 297?
Yes, actually, this analysis was done by Steve Myers at our laboratory.
And this is it has samples from Nicole Brown Simpson's dress, correct?
Yes, it does.
And the interpretation of this sample for samples G5 and G6 were an 18 allele stronger than a 24 allele.
You're referring to G3?
No, G5 and 6. Let me highlight those at the top. See G5 and G6?
Yes.
And you agree that that is an accurate picture of that film for that particular run?
Yes, it is. But once again, it's difficult to see the weaker components of this, and you'll actually have to look at the gels yourself.
All right, let me zoom in on G5 and 6. Would you agree that you cannot see any 24 allele, at least on this picture? What you see is the 18s, correct?
Correct.
There is nothing apparent here.
Well, this is all done with your computer. I mean, this isn't the actual sample, and so I don't know.
Would you look at your original sample and tell me if you agree that it is extremely faint?
It's a trace level of 24.
So this is a
But
trace, I'm sorry.
I'm sorry, but you can see it in here, and when you do see a copy of it later on, you'll be able to see that it's a trace amount of a 24 allele.
How much more is it than a hint?
Well, a hint, as I stated, is a hint is a possible. It's not a well-defined band. This trace, these are well-defined bands, and they're just very faint. And as far as why you can't see it on your monitor, I can't explain that. This is the actual
You have
copy.
I'm sorry, you have the actual one there?
Yes, I do.
We take a look at that?
Yes, but I would prefer that it not be put into evidence. Mr. Harmon has a copy of the blue copies.
Well, let me use the blue copy. Is that adequate?
Oh, yes, Mr. Harmon has those copies, I believe.
Mrs. Robertson?
Did you get that a number?
Yes, we did.
277.
In the corner. All right.
But this is Exhibit 275I. Everybody's on Elmo?
Go. Mrs. Robertson? Vote. Does it say AG 297 on that, Mr. Harris?
I believe it's just backwards or upside down.
Oh. Okay. Now, the area of interest. Now, the area of interest that we're looking at are the 2 lanes over toward the right between the 2 ladders, correct?
Correct.
And you're saying that there is something more than a hint, which is a band, at 24.
Correct.
And can we put arrows in that area, Mr. Harris? And can we print that out, please? And I'd like to have that shown to the jury, please. Mark this print out.
All right, if you'll hand, you want to hand the blue copy?
Maybe we could hand both at the same time.
All right. About 3 minutes to print that out.
Can you make the printout 1176, please?
1177.
1177.
Make it print out 1176A.
Whatever your pleasure is.
Well, do you want to keep them together?
I think we just have one. That's fine.
All right.
That's good. 1176A. let's hold off on that and show another picture. We can do a couple of them at the same time, if that's okay.
Proceed.
Could we have photo number 92, please?
Can I see a copy, that blue copy?
Sure.
Thank you.
Now, Your Honor, could this be marked as the next in order,
1177.
1177? Now, Ms. Montgomery, the picture that we've put up now, 1177, contains the samples from Nicole Brown Simpson's fingernail scrapings, does it not?
Yes, it does.
All right, with the court report, please.
Now, Ms. Montgomery, this is one of the pictures that you looked at lunch to compare with your original film, correct?
Correct.
Sustain for his question.
Well, this is a scanned image of the photograph that I have right here. Would you like to look at it?
Actually, it's irrelevant.
Oh,
Would you like to look at the photograph that you looked at to assure yourself it's the same one?
Actually, that's irrelevant.
oh.
If you would like to hand that to me, sure. And yes, this is a photograph that you asked me to look at during lunchtime, and it appears to be a photograph taken by Dr. Ed Blake of one of my D1S80 gels.
Okay, and does it appear substantially the same as the scanned photograph, the same photograph?
Actually, it's irrelevant, whether it's substantially the same.
Sustain.
Does it appear to, I'm sorry, does it appear to be the same on the screen as what you have in your hand?
Well, it has the same labeling and everything. I mean, this is a computer scan of the actual picture. So it appears similar. It's missing some of the side writing that's on these pictures.
Does it look any different at all, other than the side writing, other than the scanned image being a little smaller?
No, it looks very similar. I mean, it appears to be the same one scanned into a computer, I guess you're doing.
And you compared these photographs with your films during lunch, did you not?
Yes, I did.
And you indicated to me after lunch that these appeared to be accurate representations of your films, correct?
Correct. Some of the subtle, I mean, as I stated earlier, you need to actually see one of the blue copies to see some of the subtle or some of the less intense bands. And I assume you'll be seeing those later.
Okay. And if any of these pictures that I show you, if something else is revealed on your copy, you tell me and we'll show that your copy as well, okay?
Okay.
Now, this is the run that you did with the samples from the fingernail scrapings, correct?
Correct.
And the fingernail scrapings appear in these lanes, 45B, 46B, and 45A1, correct?
Correct. Those are the scrapings and the clippings.
Okay, which is which, just so it's clear.
45B, referring to my notes, 45B is the right scraping. 46B is the left scraping, and 45A -B1 is the right clipping.
Now, let me back out of this, and I want to highlight those 3 lanes. Now, would you agree that there appears to be something going on below the 18 allele, correct?
You know, you're focusing in on these and zooming them up. You need to look at the actual gel. You can't, we don't
let's look
image
at-
our gels. We just do a hands-on
Okay.
look at the gel.
Strike it, and I'd ask the court to take that exhibit off the screen.
No, proceed.
Let's look at your original of this one, okay?
What was the gel number again? AG 293?
293.
Yes, this is my original. I have taped to the back a transparency with the labeling on it, but it's just a temporary removable item.
And would you agree that the area that I've blown up here conforms to what's on your original in terms of these band-like items below 18?
Well, no, this is like taking a very close-up photograph and trying to ask the person what they see in the whole photograph. You need to look at the big picture. You can't just focus in like that. It's
Well, look at the big picture.
inaccurate. By looking at this gel, there's just some silver precipitating in this region, and once again, you'll be able to see these. And there's just, you know, a little darkening down here. It's not a band by any means.
Would you agree that it looks like what we have up on the screen?
Well, this is a magnification. So,
Yes.
I mean, if I were to try to magnify this with my eyes, yes, but I think you need to look at the actual data and not magnify it up like that.
Well, we'll look at it. Does it look like there is something going on below the 18 allele at the position of the 17 allele? Under 46B.
What you can see is, if you look up at the gel, you could see a smearing from the first ladder all the way, I can't see it from here, but if you look at the first ladder and all the way over to past the second to the last ladder, you'll see this dark smearing, and that's just a little silver being precipitated. That's just a smearing. It's during the staining process, and it's not by any means a band.
Would you agree that neither Nicole Brown Simpson, Ronald Goldman, nor OJ Simpson have a 17 allele in the D1S80 system?
I agree with that, yes.
Now, this smearing that you're talking about that causes that activity in the 17 allele, is this something that went wrong with this test?
No.
Is this kind of smearing something that you typically see where you have, call them band-like appearances, appear on the gels?
Sustain. Rephrase the question.
Is this kind of smearing something that's typical for the D1S80 system?
It's not, well, no, I don't typically see something like this, and it's just, like I said, the silver is being precipitated and causing a nice flowing smear through that portion of the gel.
Did you run this again to see whether that activity at allele 17 disappeared?
Well, I don't see any activity at allele 17.
Did you run this again?
No, I did not.
Well, we have the picture, so. Here, and I would ask that this particular photograph, as well as the last one, be circulated to the jury.
All right, at this time? All right.
And
1177, 1176 with the printout.
in the last one, we'll submit the blue copy as well.
All right. In 1177, Mr. Blazier, would you hand 1177 to juror number 7 and hand 1176 and the blue copy to juror number 1?
Now, the photograph that's up here, she's been referring to her original, which is different from the blue copy, and I'm wondering if the court would like me to circulate her original with my photograph, just so the jury can look at both.
May I see the original? Is this something a layperson can read by looking at it?
Oh, yes, these are. And you want to try to avoid touching the gel because you don't want to leave your oils on it.
Yep. All right, then we ought to do this in one set at a time.
Okay?
All right, ladies and gentlemen, be careful how you handle the original gel, please.
Handle on the side, because if you, just like photographs, if you touch the center, it kind of causes some residues.
So I will hand 275I and our printout, which is 1176A.
All right. We're just looking at that. Let me see, counsel, without the report. B.
I this is the gel that shows 30, 31, 293, and 305. You have that in mind? Oh, maybe it isn't.
What AG number was that?
174.
Samples. 275B is reference samples, correct?
Now, do you see in the reference sample lane for Mr. Simpson, there appears to be 3 bands. You agree?
I'll refer to my copy.
Okay. Could we zoom in on that a little bit more? Actually, there appears to be 4 bands, I think. Would you agree there appears to be actually 4 bands in that reference line?
Yes, that's a shadow band effect.
That's not supposed to happen, is it?
Well, sometimes you see it in the center section of these acrylamide gels.
That's not supposed to happen, is it?
It's not supposed to happen. To optimize, you would prefer that it did not happen. It does happen on occasion, though.
What do you mean to optimize? You would prefer it not happen? You don't want it to happen, do you?
No, you don't want it to happen.
Something went wrong with this particular gel that caused that to happen.
Objection, that's argumentative and cheating through proof. Sustained.
Did something go wrong with this particular gel that caused extra bands to show up?
Well, something didn't go wrong with the gel. Sometimes we see these shadow bands in the inside region of the gel, just in the center section. And as you can see, it is isolated to the center section of the gel. If it was an issue, the sample should be rerun. If there's a potential that these were mixed samples or something such as that, then you want to rerun these. It doesn't change any of the results of the analysis on this gel.
Is it one of the shortcomings of this system that sometimes happens that you get extra bands on there that are shadow bands?
Objection, that's argumentative.
Sustained. Raise the question.
Is that a characteristic of this system let me ask you this, how frequently does this happen?
It depends on who is doing the analysis. Different people, this is a very techniquey system. Some individuals tend to see it a little more often than others. In our laboratory, we've been able to minimize it, so we rarely see it anymore.
How often do you get it in tests that you conduct?
I'd say less than 5% of my gels would see them.
Now, would you also agree that the band on Mr. Simpson's reference sample at 24 is uneven in the sense that it's not even the same intensity across the band? Appears to be a kind of very bright on the left side of that band.
Well, there's the artifact, that little dot right on the end of the 24 band. But by looking at this copy also has that little dot, but it appears to be relatively equal intensity across the length of the well.
That little dot's not supposed to be there either, is it?
They're saying not supposed to be there. The dot is not part of the sample. No.
So it's not supposed to be there, correct?
One wouldn't want it to be there, no.
Could we have photo number 96? And I have an additional photo that will be 1178. I'm sorry? We have photo 1174 and 1178 side by side, please. Does the photograph on the right appear to be your run 299?
No, it does not.
What round does that appear to be?
294.
Okay. And is it correct that these, the photo on the right has the samples 303 and 304 from the Bronco on them? I'm sorry, it's the one on the left. On the left of the screen is the run that has samples 303 and 304.
Correct.
And the one on the right is the run that has the samples for 30 and 31 from the Bronco and 293 from the carpeting in the Bronco.
That's correct.
Now, 303 and 304, you read those as containing a very weak 18 allele, I believe. Is that correct?
Once again, I'll look up my notes. Yes, for the two Bronco samples, DNA 52 and DNA 53, there was a weak 18 detected.
Now, I'm going to try and blow up that area. And the area that we've indicated is approximately in here, is that correct?
I have a similar objection that we obliged earlier. That's a distortion.
Or will,
And here? Have I circled the right areas?
Yes, as you can see, the 18 allele is, the 18 ladder allele is, I'm sorry, yes, that is correct.
Okay, and it's a very faint band, correct?
No, it's a weak band.
Weak band. And it's a weak band on your original as well, correct?
Yes, it's a weak band.
And that would indicate a very small amount of DNA being present, correct?
Well, it's relative to the amount that's present in the whole sample, yes.
Okay. And were you able to quantify the amount of DNA present that was contributed by the 18 allele?
No, what we do is a total quant to determine the total amount of DNA. We can't tell the difference between different alleles' contribution for the sample.
Would you agree that the band intensity of the 18 bands that I've circled is about the same as the 1 nanogram standard, or maybe less? Or what is your assessment of the relative intensities?
I'd say that's it.
pull the microphone closer.
I'm sorry. There's approximately equal to the 1 nanogram, slightly less than the 1 nanogram for DNA 53.
And the intensity of that is far less than the 24 allele and the 25 allele, correct?
Well, yes, it is less than either the 24 or the 25 alleles.
Now, samples 30 and 31 are on the right picture, correct?
Yes, they are.
And you did not see any 18 alleles on those samples, correct?
For item 30 and 31, no, I did not see an 18 allele.
And I'm going to make a circle the area where you would expect to see that, specifically there. And would you agree that I've made little squiggles there about where the 18 band would be?
Yes, that proportion of that.
Okay.
Correct.
And you indicated there was nothing there that you saw that could be a band.
That's correct.
Could we have that printed, please? Now, I want you to assume for a minute that 30 and 303 came from the same general area.
Proper hypothetical mistakes the test.
Sustain.
If you had a mixed stain that had alleles present in part of the stain that weren't present in another part of the stain, do you have that in mind?
So what you're saying is a blood stain where one person's bleeding here and one person's bleeding here and some of one person's diffusion into another?
No, let's just say you have a stain, a fairly relatively large stain, and you test one area of the stain and you find some alleles. You test another area of the stain and you find some additional alleles. You have that in mind?
Yes.
Would that be an indication, would one explanation be that those stains were put there at different times?
Sustain. Sustain.
Would that be consistent with two different stains being put on that surface at different times?
I couldn't make a determination as to that, no.
Okay. Could we have 11 photo 90 back on? Now, you have no personal information about what happened between when sample 30 and 31 was collected and when samples 303 and 304 were collected, do you?
No, I don't.
Now, the appearance of the 18 allele on 303 and 304, the very faint band, would you agree that the children of OJ Simpson and Nicole Brown Simpson both would have an 18 allele?
No, that's incorrect.
Why is it incorrect?
You're saying they would, or?
They would have an 18 allele, both.
Oh, I'm sorry. You are correct. If the mother's an 18 homozygote, then the children would all have an 18 allele present.
And there's a strong likelihood that those children might also have a 24 and a 25, or a 25.
Correct.
And that's because you inherit these alleles from your parents.
Correct.
And if there were stains mixed in with a blood stain that, from the children, you might expect to see some of the children's alleles in the stain. Correct?
If you had individuals with the same type, yes, that's possible.
And you have no way of telling whether the 18 allele in samples 303 and 304 was put there or got into that sample at the same time as the 24 and 25 alleles, do you?
No, I don't.
You have no way of aging how old the sample is from looking at the gel, correct?
Well, some indication can be obtained by if a yield gel is done on a sample, you can obtain some information if degradation has occurred. But
Okay.
by looking at strictly the D1S results, no.
But you can't tell, you might be able to tell that there was degradation, but you can't tell how old the stain is, can you?
No.
Could we have, I have two more photos, Your Honor. One would be
1179 and 1180.
1179 and 1180. We have photo 97 and 98. Now, the photograph on the left, would you agree that that appears to be the gel with the samples from the glove, the Bundy glove, I'm sorry, the Rockingham glove? G11, 12, and 13.
Yes.
And the one on the right, which is photograph, which is exhibit 1180, appears to be stains G1, 2, 3, 4, 9, and 10, correct?
Yes, that's correct.
And would you agree that you interpreted those, the three stains, G10, 11, and 13, as having a weak 25 allele?
Once again, I'll refer to the report. It's a weaker 25 allele. It's not a weak 25 allele.
It's much less intensity than the other alleles on those lanes, correct?
Yes, it's less intense than the 24 allele.
And let me circle 11 and 13. Oops. Have I got those about right?
Do the second one again. Because you encroach on the band itself.
So let me circle both of them. Okay, and the 25 band is here and here for those two samples, correct?
That's correct.
And the fact that it's so much less intense than the 24 band would indicate that if there was a contribution from a 24-25 genotype, that that person's contribution is very, very small compared to other contributors.
Yes, the 24, 25, or if the 25 is, well, the 25 is the minor component, along with an 18 on those samples. And the 24, 24 is the major contribution. So the minor contribution would be less than the major contribution.
And the same is true over on the picture of the right of G10 with respect to the 25 allele, correct?
That's correct, but in that situation, there isn't an 18 allele present.
But you called a 25 that's much less intense than the 24, correct?
I called a major type and a minor type, major type and a minor allele in that particular example.
Can we print that, please? Save it and print it later? And could I have slide First slide. The three stains from the glove, G10, 11, and 13, were the only stains on the glove where you found genotypes in the D1S80 system consistent with Mr. Simpson, correct?
That's correct. Those are the three areas where a 25 allele was detected along with the 24 allele.
And this is 1173A. Could we have B, please? And the indication that you have on your paperwork is that those three stains all came from the wrist area of the Rockingham glove, correct?
That's correct.
And we have the next slide, please. You tested a number of different stains from the rest of the Bundy glove, the Rockingham glove, correct?
Correct.
And isn't it accurate that you were able to exclude Mr. Simpson as a contributor to every stain that you looked at on that glove other than G10, 11, and 13 in the wrist area?
Yes, there was no 25 allele detected on the other stains.
And there was when you received this glove, do you know whether it was turned inside out?
Gary Sims did all the examination of the evidence in this case, so I don't recall.
As a forensic specialist, if you got a glove as a piece of evidence and you wanted to turn it inside out, how do you do that? Do you have to handle the wrist area a lot to get it turned inside out?
Well, I think it's not just a forensic analyst, but how would an individual in general turn a glove inside out if it were out the opposite direction?
Would you agree it would involve considerable manipulation of the wrist area of the glove?
Yes, I would.
Sustain. It's vague.
This would be an appropriate time to break.
Finish.
Do you have any way of quantifying the amount of DNA attributable to the 25 allele in G10, 11, and 13?
No.
Do you have any way of placing an upper limit on it?
Yes, I could. By looking at the relative intensity of the 25 compared to the major contributor of a 24 homozygote, I could tell you the extremes that it can be. And for the sensitivity of our system, what we find is at beyond 1 to 20, you can't see the minor component. You typically cannot see the minor contribution in a sample.
But you have no way of quantifying how much DNA was present that caused the 25 allele to appear.
The exact amount, no.
And it could be a very small amount.
It's vague. Sustain.
Could be below 1 nanogram, couldn't it?
Of total DNA or of DNA on the
DNA on the gel.
on the gel? Well, in this case, what I could do is compare the 1 nanogram standard, the 25 allele, to the 1 nanogram standard. And would you like me to do that?
Yes.
That 25 allele is approximately equal intensity to the 1 nanogram control that's on the gel.
And I'm sorry, which sample were you looking at?
I'm looking at G11 and also G13.
How about G10?
G10 is also approximately equal to the 1 nanogram standard.
It's a much smaller amount compared to the other alleles on the gels, correct?
Yes, the other band in those samples looks a little greater than the 4 nanogram standard.
And you have no way of telling from this test whether or not that 25 allele got on that glove at the same time as the other alleles on the glove, do you?
No, I don't.
That's all I have.
All right. All right, ladies and gentlemen, we're going to take our recess for the afternoon. Please remember all of my admonitions to you. Don't discuss this case amongst yourselves. Don't form any opinions about the case. Don't allow anybody to communicate with you. Don't conduct any deliberations till the matter has been submitted to you. As far as the jury is concerned, we'll stand in recess until 9 a.m. tomorrow morning. And Ms. Montgomery, you're ordered to come back tomorrow morning, 8:45. All right? Stand in recess. Thank you.